plasmid replication, and some of them are DNA dependent. And an example of such plasmid is RK2 with its own origin of replication ORIV, with its structure that consists of sequences DNA boxes to which binds host protein DNA A and iteron sequences to which binds plasmid encoded protein TRFA. And our laboratory, based on the research on TRFA, has proposed a model of initiation of replication where where TRFA has ability to bind single-strand duo. But the advanced molecular role of DNA A protein in this process is not well known. And for that, I propose my question, does DNA A bind single-strand duo initiation of plasmid replication? So I have constructed two plasmids with single substitution for alanine in DNA A gene in place valine 211 and arginine 245. And after overproduction and purification, I have investigated the affinity to DNA. And I, for that, use method biolayer interferometry, where I use a sensor tagged with streptavidin, which I incubated with DNA tagged with biotin, and such sensor loaded with DNA, I then incubated with one of the variants of DNA A protein. And the main goal of my research were single-strand duo of top and bottom, and this is the result, where it's shown that DNA Y-type has the same affinity for both strands of single-strand duo, and variant R245A has the lowest affinity to both strands. So DNA A not only melts duo, but also participates in helicase loading, and to check this ability in for my variant's ability in this process, I have conducted helicase activity assay, where in reaction mix consists of all proteins participating in initiation of replication with my variable, one of the variant DNA A, and I have performed two sets of this experiment on ORIC and ORIV. And in the positive sample where helicase loading occurs, such plasmid molecule under the activity of helicase and gyrase migrates a little bit faster on gel, giving this lower band. So this is the result on ORIC, which shows that both variants V211 and R245 are inactive in this process compared to the Y-type. And on ORIV system, both variants retain the ability to load helicase. And here I want to point the variant R245, where it looks like it has lower efficacy in this process. So in conclusion, in contrary to the ORIC system, both variants retain the ability to load helicase on ORIV origin replication, and the possible explanation for lower efficiency for variant R245A may be due to its lower affinity to single-strand duo. But nevertheless, this further research is required to estimate the role of DNA A in this process. So thank you for your attention, and I encourage for discussion.
Thank you, Mateusz. Do we have any questions from audience? If not yet, I will happily start with my first question, which will be about the choice of the substitutions that you made. So why particularly these two? What were
thank you for your question. Ideas? So they are already publications about these both variants in the system of ORIC, where their role in initiation of replication in chromosomal DNA, and also on the molecular basis is that alanine is a small amino acid with neutral charge, so it's less likely to have any interaction.
Okay, thank you very much. I have another one regarding the tagged DNA. If you could please go to the biolayer interferometry bindings. How about the other variants of the DNA that you used? Did you see similar patterns?
So for the Y type, I have seen that for the rest of the molecule, for the control, they were showing the Y type had the lowest affinity, where my variants had much higher on level comparable to this. And I don't know why I was getting such a low results for the Y type, and I have no explanation for that. Maybe it's because both sets of experiments on this DNA molecule and the others, I was using from different elements of DNA Y type.
Okay, thank you. Any other questions from the audience? If not, thank you very much. And I am
analysis of the effect of thymoquinone on the stability of cholecalciferol using the HPLC technique. My supervisor is Dr. Leszek Gdanski, and I worked in laboratory of physical biochemistry. Let's start with a short introduction. Cholecalciferol, or vitamin D, has multiple beneficial effects in humans. It aids in calcium and phosphorus absorption, helps with immune function, and in regulation of cardiovascular functions. Because of that, it's often recommended to supplement diet with vitamin D3 or cholecalciferol preparations. However, those are prone to oxidation and degradation, which leads to use of stabilizers for their preservation. Those stabilizers are often artificial or require high concentrations to function. I've tested thymoquinone, found, for example, in curcumin, as a potential natural stabilizer for those preparations. Moving on to aims of my study, I wanted to develop and validate an HPLC method for determination of cholecalciferol concentration and to analyze the effect of thymoquinone on the stability of cholecalciferol. To do this, I've used the following methods. First, I've prepared the stock solutions, which I've diluted with oil, incubated in various temperatures, then diluted with mobile phase for HPLC analysis. To be precise, I've used a normal phase HPLC variant, which, contrary to its name, is much less popular than the reverse phase HPLC. The difference between the two is that normal phase uses non-polar mobile phase with polar and hydrophilic column, and the reverse is true for the reverse phase HPLC. Moving on to method section, results section, we have method development. I've tested 2 HPLC columns along with 3 different solvent additions in multiple concentrations. At first, we've worked with monolithic column, which you can see here. Its internal structure is filled with numerous channels, and it's intended for tight and sharp peaks in chromatograms. Next, we've tested a standard particulate packed column and to compare the two. Here you can see the comparison of peak shapes with the addition of isopropanol. On the left are the results from monolithic column. Here, as you can see, the baseline is incredibly jagged, and the peak of vitamin D has a big slope at the leading edge. On the contrary, in the particulate packed column, baseline is almost completely smooth, and peak shape has drastically improved. Because of that, we went with this method for next experimentation. Those are final parameters we've used. Mobile phase consists of 99% N-hexane with the addition of 1% isopropanol. Here you can see the new peak belonging to pre-vitamin D3. It's another active form of cholecalciferol, and together they allow the determination of total vitamin concentration in a given sample. Here are results of method validation. We've tested our method for specificity, linearity, accuracy, repeatability, reproducibility, stability, and robustness. And our method passed all of those criteria. So let's move on to the actual results. Here is the comparison of peak areas after the 4-week incubation. Up the top is the sample with BHT, which is a common artificial stabilizer. And right next to it is untreated control sample with only vitamin D. And here is how they compare to samples with thymoquinone. As you can see, it not only does not stabilize the vitamin D, but degrades it, and with higher thymoquinone concentrations, this degradation process is more noticeable, with around 10% difference between 10 micrograms and 200 micrograms. The same is true when we look at the comparison in multiple temperatures after the 4 weeks. But here you can see that the effect is smaller in lower temperatures, with around 5% difference maximum between 25 and 40 degrees. Now let's move on to summary and conclusions. So, vitamin D3 or cholecalciferol preparations require stabilizers for preservation. These are often artificial or require high concentrations to function. Thymoquinone does not stabilize the vitamin D3 and instead degrades it. And with higher thymoquinone concentrations, this process is accelerated. These findings are important because there are already supplements on the market that contain both thymoquinone and the vitamin D3. Not only that, they often lack thymoquinone concentration labeling, which could result in even higher loss of vitamin D3. With that, my presentation ends, and thank you for your attention.
Thank you very much, Paweł. Do we have any questions from the audience? Yes, I will come with a microphone.
So I think on the first method slide you show that you diluted the samples in oil, and I would like to ask if it was any specific kind of oil or just any...
That's a good question. Could someone up top help with a little more? A little more? A little more? Here. I think this slide will be best to showcase this. So we've used Magiol 812, which is a MCT, or medium chain triglyceride. And we chose it because it's a non-toxic, inert oil with little to no impurities, which means that it doesn't affect the detection methods in HPLC and doesn't cause any degradation in temperature incubation because it's highly stable. And in contrary to, for example, cooking oils, it provides reliable results that are easy to reproduce in other systems.
Okay, thank you very much.
Thank you for your question.
Thank you. Do we have any other questions from the audience? If not, I have just one more regarding the artificial stabilizers of vitamin D3 that are currently used. So do you have any ideas if these currently used can be, for example, toxic, or why is there a need to replace them with some others?
First of all, they could be toxic in extremely high concentrations. Preparations itself are generally safe, but it would be better to switch to a natural alternative. However, currently, only one that I know which is used is vitamin E, which, however, needs around 53% concentration by volume to work as a stabilizer. And that's why we've tested thymoquinone, which was supposed to work both in low concentrations and, well, work.
Okay. Thank you very much.
Thank you.
Hello, my name is Aaron Gorbacz, and today I will present you my master thesis titled Influence of Polyethylene Glycols on Bioencapsulated Beta-galactosidase, Modeling and Optimization of Composite Synthesis Process. It was done on the Laboratory of Physical Biochemistry under the supervision of Dr. Leszek Gdansk. Xerogels are porous materials obtained by the Soldier method. They are characterized by the high porosity, low density, and high surface area. Their name comes from the Greek word xeros, meaning dry, as they are obtained by drying in ambient conditions. But the final product of the Soldier method may alter depending on changing of the conditions of the drying. Xerogels are synthesized from precursors called alcoholants. They contain a group called alkoxysilans, which with the addition of water undergo hydrolysis, forming silanol and alcohol. Silanols then under condensation react either with each other, forming siloxane and water, or cross-react back with alkoxysilane, forming siloxane and alcohol. Siloxane is mainly responsible for the xerogel's porous structure. Xerogels find various applications due to their unique properties, ranging from drug delivery, thanks to the non-toxicity to humans and high porosity, to window insulation, thanks to the thermal conductivity and ability to preserve energy. Its one prominent application lays in catalysis and bioencapsulation. Thanks to its high porosity, it provides high accessibility for the enzyme's active sites. So in my work, I decided to bioencapsulate beta-galactosidase. Why beta-galactosidase? Well, it's a perfect enzyme for experimentation due to the fact that it's well characterized and easily detectable thanks to its synthetic chromogenic substrates such as XGAL or ONPG. Beta-galactosidase is mainly responsible for lactose hydrolysis. thus finding use both in science and food processing due to the increasingly common lactose intolerance. The goals of the study were to successfully bioencapsulate beta-galactosidase in silica composites, to model and optimize such composite synthesis process, and to study polyethylene glycol's influence on bioencapsulated beta-galactosidase's physical-chemical properties. And in order to achieve this, I used design of experiment statistical models, which allowed me to model my probes in a way that required the least of my resources but provided me with the most amount of data. I used Box-Benken statistical model, which modeled me 13 probes with polyethylene glycol's concentration ranging from 1 to 20% of various molecular masses. After I synthesized such probes, conducted enzyme activity assays, and after a statistical analysis, I have synthesized a perfect optimized gel according to design of experiment. And then on this exact gel, I have performed future enzyme activity and stability assays. Going to the results, first thing after gel is synthesized, it is crushed, sieved, and sorted by size. And in order to check whether the particle diameter had any impact on beta-galactosidase's activity, I conducted an enzymatic activity assay using ONPG, which after hydrolysis produces ONP, which is a chromogenic product measurable by absorbance spectroscopy. The results indicate that there was no big difference between activity in particles diameter ranging from 1 to 400 micrometers, but the particles with the diameter over 400 micrometers showed a significant activity loss coming up to almost 60%. Thus, in every future experiment, I have used particles not exceeding the 200 micrometer diameter. Design of experiment statistical analysis showcased an F value of almost 200, meaning that the model was very significant. An F value this high could only occur in 100% chance due to noise. But the polyethylene glycol's concentration didn't have any statistically significant impact on beta-galactosidase's activity. As seen on this graph, with the addition of polyethylene glycol, the activity of beta-galactosidase doesn't change slightly. When it comes to polyethylene glycol's molecular mass, on the other hand, the graph is pretty steep, signifying that the lesser the molecular mass of the polyethylene glycol, the higher the activity of the beta-galactosidase. Thus, according to design of experiment, I have synthesized a perfect gel consisting of 40% of polyethylene glycol's concentration of 200 Dalton molecular mass. On this exact gel, I have performed every future experiment, starting by thermostability. I have conducted the same activity assay using ONPG in temperature spectrum. I subjected both gel with polyethylene glycol and without polyethylene glycol to the experiment in temperatures ranging from 25 to 80 Celsius, and both of them have showcased the highest activity in 60 Celsius. But although the activity was pretty similar, the xerogel containing polyethylene glycol showed a slightly bigger activity in both lower and higher end spectrums. The next thing I have changed is composite reusability. I subjected one exact probe to the same enzymatic activity assay to 11 consecutive reactions and measured its activity. The beta-galactose's activity loss in continuous reaction was exponential. It showed almost 60% activity loss after 3 reactions, up to almost 80% after 9 reactions. But despite losing 80% of its initial activity, the composite proved to be reusable as it maintained a steady course after probe number 9, not losing any more activity. So summarizing, I have successfully bioencapsulated beta-galactosidase in xerogel. Polyethylene glycol's molecular mass has statistically significant impact on bioencapsulated enzyme, and the addition of polyethylene glycol has proven to slightly improve the enzyme's thermostability. Thank you for your attention.
Thank you very much, Aaron. Do we have any questions?
I just didn't catch. You were using any statistical software or for preparation?
Yes, I have used software called Design of Experiment, which modeled all of my probes and then conducted the statistical analysis. And the statistical analysis has shown that polyethylene glycols concentration didn't have any factor in beta-galactosidase activity, but polyethylene glycols molecular weight had a significant impact on beta-galactosidase activity. Thus, it kind of showcased me what kind of gel with these factors would show the biggest activity of the bioencapsulated beta-galactosidase.
Can you remind me the name of the software, because I didn't catch it at the beginning?
I'm not exactly sure. It was done on the laboratory computer. I just know it's Design of Experiment. I'm sorry.
Okay.
I have no significant, I don't have enough knowledge to answer the question.
Okay, thank you.
Do we have any other question from the audience? If not, I have just one last. You mentioned that the xerogels can be used in drug delivery. So I'm actually interested, what happens with the xerogel itself after it's introduced to the body? Is it like soluble, or is it excreted in the full xerogel form?
Well, according to a work done on Gdansk Medical University of Gdansk by Dr. Czernowy, it has the ability to form a hydroxyapatite, which is basically a layer within the bone. So it stays there.
Oh, that's interesting. Thank you very much for your talk. Thank you.
Thank you very much.
Marceli Bogusławski with his talk titled Interaction of doxorubicin with platinum nanoparticles with diameters of 5 and 70 nanometers. The floor is yours.
Welcome. I am Marceli Bogusławski, and this is my presentation about my progress of my project, master project. And its title is Interaction of doxorubicin with platinum nanoparticles with diameters of 5 and 70 nanometers. And let's start with introduction to doxorubicin. It's antibiotic from anthracycline group, first isolated from Streptomyces paucitius, and it's found its use in treatment of different cancers, such as breast cancer or leukemia. On the right side of the slide, we can see its structure, which is 4-ring aglycon connected to downazamine. When it comes to the action of doxorubicin, it's mainly focused how it kills cells. It's mainly focused on the intercalation into the DNA, but it can also inhibit topoisomerase II, and it can create reactive oxygen species. And although doxorubicin is regarded as one of the most crucial chemotherapeutics due to its efficacy, it is associated with several side effects some of them I listed here on the right side, but the most important one are being cardiomyopathy and nephrotoxicity. And to minimize those side effects, researchers propose the use of combined treatment with other drugs and the use of advanced delivery platforms. And one of the approaches that could be possibly can be a good answer, yes, is the use of metallic nanoparticles. And I will be, in my research, also using platinum nanoparticles that I commercially acquired from nanocomposites, and 5 nanometer with 2 different diameters. 5 nanometer were suspended in aqueous 2 millimolar sodium citrate, and 70 nanometers were suspended in aqueous 4 millimolar sodium citrate. And my research goals, can I have the next slide? research goals were to investigate whether doxorubicin interacts with platinum nanoparticles. Are there any differences in those interactions depending on the size of the nanoparticles? And if those interactions influence the mutagenicity of doxorubicin? And I started my methodology with spectrophotometry measurements. And on the left side, we can see controls when all spectrophotometry measurements were measured during the titration of doxorubicin with platinum nanoparticles. And on the left side, is control where I, during the titration, replaced nanoparticles with buffers that the nanoparticles were suspended in. And as we can see, the molar extinction didn't change in this case, but with increasing concentration of nanoparticles, it did increase. And similar situation can be seen during the fluorescence measurement. Here, as controls are the same, and here we can see a slight decrease of the fluorescence due to decreasing concentration of doxorubicin, but the scale of this event is much more visible during the titration with nanoparticles. And I also performed dynamic light scattering, and in the measurement where I measured 5 nanometer particles with nanoparticles with alone, we can see two peaks here and here. And the second one can be explained by the tendency of those nanoparticles to self-aggregate. After the addition of doxorubicin, the hydrodynamic diameter changed, and we can see a homogenization of this sample. And after the addition, the hydrodynamic diameter further increased, meaning a high level of aggregation. In case of 70 nanometer particles, after the addition of small concentration of doxorubicin, the hydrodynamic diameter didn't change. And to achieve the increase, I had to add additional doxorubicin. I also decided to measure the enthalpy change using the isothermal titration calorimetry. And here are titration thermograms that allowed me to plot those graphs. And after the extrapolation to 0, I achieved that the values of delta enthalpy change. And as they indicate that the interaction between 5 nanometer platinum nanoparticles and doxorubicin is exothermic, and between 70 nanometers and doxorubicin is endothermic. And unfortunately, those differences can be explained by the use of different buffers that the nanoparticles were suspended in. And finally, my only biological experiment, I performed 2 tests. In both cases, I used the same controls. Negative control was only water, and positive was doxorubicin. Here I checked whether nanoparticles have a mutagenic effect on Salmonella, and it showed that they don't. And here I mixed nanoparticles with doxorubicin. Unfortunately, only one result was statistically significant, and the result shows that nanoparticles in small concentration decrease the mutagenicity of doxorubicin. And in summary, doxorubicin and nanoparticles indeed interact with each other. There are small differences in the interaction of doxorubicin depending on the size of the nanoparticles, and doxorubicin platinum nanoparticles interaction have influence on mutagenicity. And thank you for your attention.
Thank you very much for your talk, and I would like to ask for some questions from the audience.
Hello. I have a question about the different concentration of this citrate buffer when it comes to the 5 millimeter and 70 millimeters particles, because in one I think was 2 and in the second was 4, right?
Yes, right.
Nanomolar, I think. So what is the
I mean,
difference?
the difference here is due to the stability of those nanoparticles in buffer. Usually, nanocomposites sells them for researchers that only research only on one size of the particles. So that doesn't mean. I was thinking about trying to resuspend those nanoparticles, but that would damage the homogeneity of the sample, I suppose. Those small nanoparticles have a tendency to self-aggregate, and that would impact the results of the experiments.
Okay, thank you so much.
Thank you. Do we have any other question from the audience?
So hi, I have a question. Why did you choose 5 nanometers and 70 nanometer particles?
The question is pretty simple because I just wanted to try the smallest one available and the biggest one. That's the answer, to be honest.
Okay, thank you.
Thank you very much. Any other questions? If not, thank you so much for your presentation.
Yeah? Oh, okay. Yeah, it's responsible for the most recent pandemic, and it has a large genome with almost 30,000 nucleotides. And I will be focusing on the non-structural proteins. There are 16 of non-structural proteins, and my main interests are NSP3 protein and NSP4 protein. And double membrane vesicles are induced in cells infected with a number of positive sense RNA viruses. They work as viral factories, providing a safe and closed environment during the viral genome replication process. And creation of DMVs during the SARS-CoV-2 infection goes as follows. After translation of the first open reading frame, the polyprotein is produced, and then NSP3 and NSP5, which has proteolytic activity, cleave single NSPs from the polyprotein. Transmembrane proteins, NSP3 and NSP4, cause the endoplasmatic reticulum membrane to curve, and this curvature is
causing double membrane vesicle formation with the viral genome replication going inside. Antigen presentation is a very important process in our body, and I would like to draw your attention to the endoplasmatic reticulum's role in this process, as it is the place of a transportant associated with antigen processing and also loading peptides on the MHC molecule. So the changes that DMVs introduce into the ER may disrupt the antigen presentation process, which is the hypothesis of my research. And in my research, I want to generate the cell lines model for DMV formation and further research. For this purpose, I am using Meluso human melanoma cell line, which is expressing MHC class 1 and class 2 molecules. And I want to generate MJS NSP3 cell line, MJS NSP4 cell line, and MJS NSP3 NSP4 cell line. Currently, I am in the process of analyzing MJS NSP3 NSP4 cell line and MJS modified NSP3 NSP4 cell line. I already obtained both NSP3 and NSP4 plasmids. I also had to modify NSP3 plasmid, which I will tell you about in a minute. And I already generated MJS NSP4 and MJS NSP3 cell lines. Here you can see my Western blots that are confirming the presence of NSP3 protein in MJS NSP3 cell line and NSP4 protein in MJS NSP4 cell line. And I also used proteasome inhibition. And as you can see, there are no significant differences, which means that the proteins are quite stable in the cells. I also confirmed NSP4 expression via intracellular staining and flow cytometry, and my MJS NSP4 cell line after the sorting had almost 90% of positive cells. Yeah, and the plasmid modification, I used the commercially available plasmid, which had a BioID2 domain, which is not necessary for my research, and in fact it may disrupt the formation of DMVs. So I did the restriction analysis to remove the domain, and here you can see the gel with 5 clones I obtained, and after DNA sequencing I decided to use the clone number 4 for my further cell line generation. And my upcoming plans are comparison of the MHC and class 1 and MHC class 2 level between the cell lines, imaging DMVs using the microscopy, and tap transport assay. Thank you for your attention.
Thank you very much for your talk. Do we have any questions from the audience? If not, I will start with, I have 2 questions. First is it only the ER that the virus can use, or are there any other organelles or intracellular structures that the virus can use to obtain the membranes?
The SARS-CoV-2 DMVs are formed in the endoplasmatic reticulum membrane, but other positive sense RNA viruses can form DMVs from other intracellular membranes, mostly in the ergic region, and I think Golgi apparatus too, but I am not sure.
Okay, and another one, so when it forms these DMVs from the ER, are there any other, let's say, physiological functions of the ER also disrupted, or is it just the MHC pathway?
I think that it may disrupt other cellular functions, but the cell is functioning enough to produce viral particles. So it's not like the cell is completely frozen and like the processes are still ongoing in the cell.
Okay, thank you.
Thank you.
Do we have any question? If not, thank you very much for your presentation.
Good morning. My name is Pola Szolarowska, and today I would like to present my master's thesis titled Development of a Model for Structural Studies of Viral Proteins and the Antigen Presentation Pathway, conducted in the Laboratory of Virus Molecular Biology with Dr. Andrea Lipińska supervision. The antigen presentation pathway connected with MHC class 1 involves processing of intracellular proteins, for example viral proteins, into peptides that are then transported by the TAP transporter into the endoplasmatic reticulum, loaded onto MHC class 1 molecules that are then displayed on the cell surface for the recognition by the cytotoxic T cells that can eliminate infected cells and stop infection spreading. Some viruses evolved to inhibit or even degrade certain parts of this pathway. TAP transporter, being the central part of the antigen presentation pathway, exists in three main conformations: inward open, intermediate, and outward open conformation. Currently, the only obtained structure of this transporter is in inward open conformation. In this picture, we have the first ever obtained structure of the TAP transporter, trapped by ICP47 protein inhibitor from the herpes simplex virus. This inhibitor is additionally stabilizing this complex. The structure was obtained in the Rockefeller University using cryo-electron microscopy. In my work, I'm interested in another viral inhibitor, UL49.5, from bovine alpha herpes virus 1, that is able to hijack the cellular degradation machinery via KLHDC3 to induce TAP degradation. If we want to obtain the cell line stably expressing this complex, we have to delete the KLHDC3 protein as a part of ubiquitin ligase, for example using CRISPR-Cas9 deletion that was already done in another cell line, and now we want to introduce it into the human embryonic kidney cells without complex and glycosylation. During the deletion with the CRISPR-Cas9, there are two main steps. First is designing of the single guide RNA complementary to the gene of interest, and then delivering this complex into the cells that already contain Cas9 nuclease to disrupt the gene, leading to the gene deletion. In the big picture, we would like to determine the structure of TAP in the intermediate conformation, trapped by UL49.5 protein. And this complex will be purified from HEC-GNT minus cells. Those are the cells that lack complex and glycans. And my aims for the master thesis is to test antigen presentation in HEC-GNT minus cell line, to construct cells expressing Cas9 nuclease as a model, and to adapt this model for TAP UL49.5 studies. And going into the results, when we are comparing the MHC levels between HEC293 and HEC-GNT minus cells using flow cytometry, we can clearly see that the latter have significantly more MHC1. It's important to note that here we tested only the surface level MHC1. And we can draw similar conclusions looking at the Western blotting, comparing TAP transporter levels between those cell lines. In HEC-GNT minus, we have more TAP transporter, and this is indicated by the stronger band. We can draw additional conclusions that in HEC293 293 containing UL 49.5, there's a clear tap degradation. Could somebody change the slides for me? Thank you. And here's the main result of my work, which is the model, HEC-GNT minus cell line with Cas9 nuclease. The Cas9 nuclease was tagged by the blue fluorescent protein. That's why we can easily observe the cells containing this nuclease with using of flow cytometry. On the left panel, we can see non-sorted cells. Mostly those are the cells without Cas9 nuclease. That's why we decided to sort the cells and perform clonal selection. Briefly, clonal selection is the process when we are performing serial dilution of the cells so that we can obtain one cell per well. Those cells are then dividing, creating the homogeneous population, and I was able to obtain those in the F3 and F6 well. In case of F3 clone, there's still a mixture of cells with and without Cas9 nuclease. But in case of my F6 clone, in 98% of the cases, cells contain Cas9 nuclease. And this is our model. Now we can introduce single guide RNA into those cells to delete the chosen ubiquitin ligase and stabilize the complex that we want to study structurally. In my case, it was, of course, KLHDC3. I introduced the single guide RNA into my model. Then I delivered UL 49.5 and sort my cells. And now I will be doing Western blotting to confirm tap recovery after KLH-DC3 deletion. In conclusion, HEC-GNT minus cells express significantly more MHC1 and tap compared to HEC-293, which could be useful for structural studies. And the generated cell line can be a universal model to introduce deletions, improving protein production for structural studies. Here are my sources, and thank you very much for your attention.
Thank you very much for your presentation. I would like to ask for some questions from the audience.
I would like to ask, why did you use the HEC-GNTI minus cell line?
We chose the cell line because of the recent publication from the Rockefeller University group. They were using the cell line in the sample preparation step to obtain the tap transporter for structural studies, and that's why we wanted to test the cell line for our studies.
Okay, thank you very much.
Thank you.
Do we have other questions? I would like to ask about the deletion of this ubiquitin ligase. So, do you know how important is it to the cell, and what are the other targets of this ligase? So, if you make a knockout, then what will be the collateral damage to the cell?
This is the multi-component ligase. That's why we can delete only the one component of this ligase, and the other targets still can be, the ligase still can target the other proteins. That's why it's important to choose the target that is not lethal for the cell. I don't know many more important targets of this particular protein, but this is one of them, and this is not lethal for the cell to delete this part.
Thank you very much. Do we have any other question? If not, thank you.
Thank you.
And now I will introduce our last speaker of this panel, Weronika Kłopotek-Główczewska, with a talk titled Analysis of Neutralizing Properties of Mouse Sera After Immunization with Chimeric HBV SARS-CoV-2 Virus-like Particles.
So, good morning everyone. My name is Weronika Agnieszka Kubota-Gubczewska, and this presentation is about my master thesis project, which is titled Analysis of Neutralizing Properties of mouse sera after immunization with chimeric HBV SARS-CoV-2 virus-like particles under the supervision of Dr. Katarzyna Grzyb of Laboratory of Virus Molecular Biology. The main subject of my thesis is SARS-CoV-2 and its protein spike. This virus caused many cases of COVID-19 since the recent pandemic outbreak with many fatalities. It contains 4 structural proteins, among them spike protein, which receptor binding domain is responsible for binding to host receptors and is the target of the neutralizing antibodies, which should be elicited by a reliable vaccine. It is considered a BSL-free pathogen. In the face of the recent pandemic outbreak, many novel vaccines, including mRNA-based ones, were introduced, but unfortunately, very quickly new variants emerged, carrying multiple mutations in spike protein, and studies have shown a significant reduction in neutralization of those new variants, and in some cases it was very significant. Even though new versions of vaccines were released, updated vaccines, were released, the need of a universal vaccines against present and yet to emerge variants of SARS-CoV-2 became apparent. A crucial method is the neutralization assay due to its ease with adjustments and variety of platforms, including especially lentiviral pseudoparticles and pseudoviruses, which lower the biosafety level to 2. Those assays have many applications connected with neutralizing antibodies, like estimating the level in patients sera after the disease, or estimating the neutralizing properties of antibodies elicited by vaccine candidates against desired variants. In the previous master thesis, mouse sera were obtained from mice vaccinated with conserved epitopes of S2 domain inserted into the protein of HBV, hepatitis B virus-based virus-like particles, but then their neutralization potency was not evaluated. At the beginning of every research in a laboratory, it is important to adjust the necessary protocol and standardize reference variants. Neutralization assays can be performed using lentiviral pseudoparticles, which in this case resemble lentivirus with spike protein instead one of the glycoproteins, or alphavirus-based pseudovirions, which contain all structural properties, all structural proteins of SARS-CoV-2. Furthermore, this thesis aims to establish the neutralizing properties of mouse sera, which were obtained during the previous set of experiments using a BSL-2-suited method, which allows easy replacement of the immunogen sequence with the sequence of the desired variants. The set of experiments followed as presented. Firstly, I obtained pseudoparticles with different variants of S protein, comparing the transfection reagents and studying the protocol modification, their influence on effectivity. Later, with what I learned previously in mind, I estimated the optimal dilution of pseudoparticles for neutralization assay by using luciferase assay. Finally, I performed neutralization assay for both pseudovirus and pseudoparticle system, first on reference neutralizing antibodies and later on mouse sera from the mice vaccinated with previously mentioned VLPs. The production of pseudoparticles, lentiviral pseudoparticles, requires plasmids expressing lentiviral genes, plasmids with genes of the desired variants of spike protein, and plasmids with lentiviral backbone with 2 marker genes, ZS-green and luciferase. Later, those pseudoparticles can be used to infect cells. In this case, it is HEC293, ACE2+, the MPRSS2+ cells, which are permissive to the virus due to the overexpression of those 2 receptors. And the transfection reagent used, lipofectamine 3000 or jet prime, does not influence the overall result of the infection. GFP enabled the visual inspection under the fluorescent microscope whether the cells produce the desired particles. But the cumulative point of the step was to analyze the lysates of cells used to produce the pseudoparticles via Western blot with spike anti-RBD antibodies. Luciferase assay I used to estimate the titration which should be used during the neutralization assay. And the luciferase assay begins with seeding cells, HEC293 with receptors. On the next day, infection with serial dilution of pseudoparticles is performed. After 2 hours, medium can be changed, and after another 48 hours, luciferase substrate should be added and luminescence can be measured. This medium chain step is optional because the infection time does not influence the outcome of the luciferase assay. This graph presents the results for serial dilutions of pseudoparticles, which I used to estimate for neutralization assay, and the dilution of pseudoparticles for neutralization assay is defined by the RRU result for negative control background times 1000. In this case, the line is drawn in the 60,000 RLU, and the points at which this line crosses with a trend line for every variant individually is the optimized dilution of pseudoparticles, which should be used during the neutralization assay. Neutralization assay is very similar to luciferase assay with one additional but crucial step, incubation of previously defined amount of pseudoparticles with antibody serum for an hour, and after this time, the mixture should be placed on the cells. Here, the medium chain step is also optional because the infection time does not influence the result. The same can be seen for the concentration. Even if it's 3 times more, it still does not influence the result. Finally, these are the results for neutralization assay for spike neutralizing antibodies. I use commercially available MM43 and R001, which neutralize Wuhan and Delta, but not Omicron, and MM45, which neutralizes all of the variants with a lower efficiency, but it encompasses the elusive Omicron variant. Finally, I tested the neutralizing properties of mouse sera S21 and S78, which are those two conserved regions of S2 and control, and it turns out that those mouse sera show no neutralization against all tested variants. To sum up, during my project, I was able to obtain antiviral pseudoparticles with different variants of S protein. Those pseudoparticles can be used as a BSL-2 laboratory substitute for wild-type virus, including variants like Delta and Omicron, as their results correspond to the expected values for commercially available neutralizing antibodies. MM43 and R001 neutralize Wuhan and Delta, but not Omicron, and MM45 neutralizes all of the variants. Further, finally, I used those pseudoparticles to test the neutralizing properties of mouse sera, which were not neutralizing. And as for my future plans, I still have to perform the neutralization assay for the pseudovirus system and compare it to the lentiviral pseudoparticles. Thank you for your attention.
Thank you very much, Veronika, for your presentation. And do we have any questions from the audience?
About this neutralizing activity of antibodies. So what now, if you don't observe the neutralizing activity? Can such a vaccine candidate be processed further? Do you expect that, for example, the neutralization can be obtained, for example, in human body? Do you think that this is the system of your pseudoparticle production that affects, for example, the structure so that you don't observe the neutralizing activity? Like, comment on the future of those constructs.
So generally, though this vaccine candidate is based on those two conserved epitopes of S2, and conserved epitopes are generally not very immunogenic. So unfortunately, it turns out that those sera were not, those mice did not produce any neutralizing antibodies, which is why it shouldn't be not further used. Because even though mice is not, of course, not a human, but it still is a strong indication that it could also happen in human. Is that did I answer everything?
Thank you. Do we have any other question from the audience? If not, if you could just comment on the main downsides of using this VLP approach, I mean virus-like particles approach in these studies. What are the main downsides as compared to the virus, the SARS-CoV-2?
Generally, I was not working on VLPs. I was working on pseudoparticles. It was obtained the previous master thesis, and I just used those mouse sera. But I believe VLPs generally contain only these epitopes of SARS-CoV-2, which is it's a good way to display definitely this immunogen sequence, and this is the target sequence that we want to have antibodies against. but it's still maybe not perfect, I believe, basically. Yeah.
Okay, thank you very much. If we don't have any other question, thank you. And this will be everything in this panel, and I would like to invite you back at 11:20, and now we have a coffee break. Thank you very much.
hear me? Welcome back to the second session of this conference. The second session will be mainly, or a lot will be about plants and microbiology and plant biochemistry, but we also have at first some speakers from the biomolecular system simulations and from the laboratory of biologically active compounds. And if everything is fine, then I would like to introduce the first speaker, which will be Hubert Mielczuk. And he's from the laboratory of biomolecular system simulations and is talking about designing new substances against antibiotic-resistant strains of anthrax. Thank you so much. Kliknąć trochę więcej. Trzymasz tej. One moment. O, teraz. Dziękuję bardzo.
Good morning, everyone. My name is Hubert Mielczuk, and the title of my master's thesis is Designing New Substances Against Antibiotic-Resistant Strains of Anthrax. The work was done in the laboratory of biomolecular system simulations, and my supervisor was Professor Raimund Kazimierkiewicz. The aim of my study was to develop new low molecular weight substances against anthrax strains that show resistance to antibiotics, which could lead to developing new therapeutic strategies to combat anthrax infections. Additionally, this work could help in the future to potentially continue the experiment with the synthesis of the compounds in the laboratory. The choice of a therapeutic target for my work was receptor TM8. So far, two different cellular receptors for anthrax toxin were identified, TM8 and CMG2. CMG2 has a higher affinity for binding anthrax toxins than TM8, but TM8 receptor occurs less frequently in the body than CMG2, which means possible reduction in potential side effects. And on the right, we have a graphic representation of TM8 homo 6-mer structure. The work plan for my project. At first, I had to prepare the input file. Then I had to minimize the energy in the system. Then I proceeded to detection of binding sites. Then to these binding sites, I have constructed my ligands. Then I have performed the characteristics of built molecules. Then I have estimated their toxicity. And I have docked the built ligands to the receptor. Here are results of the energy minimization step of the structure. On the left, we have a monomer of TME8 structure solvated in water molecules with 5 sodium ions for electric charge balance in the system. And on the right is the chart representing the decrease of energy in the system in the function of time. As you can see, the energy in the system has dropped significantly in the time of steps. Here is the graphic presenting detected active sites. It was done in the software Lake Builder version 3. And there were detected 16 active sites on the TME8 receptor. This one is the chosen for this project, as I have chosen only one. The light blue area is the area of the active site. And the pharmacophore model, which describes the interaction of the ligand with the receptor on molecular basis. The blue spheres are hydrogen bond donor centers. The red spheres are hydrogen bond acceptor centers. And the light blue sphere is hydrophobic center. Here are 10 examples of built ligands by the program Lake Builder. And out of more than 1,000 molecules selected by the program, divided into 51 clusters based on their similarity, I have taken only one ligand from every cluster, the best one. Here are the example results of characteristics of the molecules. For example, this here is ligand number 2. And the most important things are right here in the red boxes and on the right. The molecular weight of my built ligand, number of rotatable bonds number of H-bond acceptors, number of H-bond donors. And here is a table representing drug likeness of a built ligand. And as you can see, there are no violations across these five filters, which means it has a high probability to be drug-like. Here is a table of estimating of ligand toxicity by many softwares. The tested traits were cytotoxicity on mouse fibroblast cell line, carcinogenicity, mutagenicity, acute oral toxicity, toxicity in honeybees, biodegradation, and half-life in the body. And as you can see, the ligands with no violations were 7, 41, 47, 48, which correspond to their cluster number. Here are the results of docking of the ligands 7 and 41. And the interaction is shown by the yellow dotted line. And as you can see, the key amino acid residues for ligand 7 were arginine 44 and phenylalanine 8. And for the one ligand, apart from 44 and 8 amino acids, there were also a cysteine 185 taking part in the interaction. And here are the results of the docking of ligand 47 and 48. And the key residues for the 7 were lysine 183 and serine 179. And for the ligand 38, the key residues were threonine 106 and serine 108. And my conclusions, I have designed 4 promising ligands to the TME8 receptor, which could be used in treating infections caused by anthrax. I have identified key amino acid residues taking part in ligand-receptor interaction. There is a possible continuation of the work by synthesizing and testing the molecules in vitro and in vivo. But caution needs to be taken as there is some possibility of non-specific ligand binding. And I thank you for your attention.
Okay, thank you very much. Interesting presentation. Are there any questions from the audience? Just give me the microphone of yours.
Mm-hmm.
You get a microphone, don't worry.
Okay. Could you say something more about this cellular receptor? So, is it a membrane protein? And if it is a membrane protein, you do not take into the consideration the membrane presence in your predictions. So, would the membrane affect the structure? And is there a risk that this small ligand can trigger the receptor?
So it is possible that that the ligand might trigger the receptor binding, but the receptor TME8 is a membrane one. It is also known for its characteristic because TME8 stands for endothelial cancer marker. So it's also a cancer marker.
Okay. So do you think that your model is accurate if you don't take membrane into consideration with the modeling? Like it was a quite small domain. So is it the whole receptor that you used for?
It's actually not the whole receptor.
Okay.
I have taken only the monomer of the receptor out of the homo 6-mer structure, and it could be less efficient in the holistic approach to the organism because of the membrane, other proteins and membrane construction. But there are not so many tools to predict this state.
Okay, thank you. Any other questions from the audience? I think there's one question.
Maybe I missed this part, but how actually did you do the modeling of those structures? So is it like the program does it, or is it based on some kind of database of already available structures?
So there is a library of possible chemical compounds that were shown in many previous works, and the program builds to this active site, and particularly the pharmacophore. So, for example, in the spatial construction of a ligand, there is a high probability that here will be a hydrogen bond acceptor, hydrogen bond acceptor center. So the program takes my structure right here, and and to this active site, it builds the ligand itself out of the library of possible chemical compounds, because there is theoretically infinite possible explanations for the problem, but in real world, chemical compounds can only be matched in finite ways. So there are no infinite ways, and this way the program can build these ones.
Okay, I think that's it, because we have to continue with the next speaker. Thank you very much once more, and you can get your presentation there. Okay, if there are any more questions, you can ask the questions later. I would like to introduce
Today, I'd like to present to you the results of my master's thesis called Designing New HIV-1 Reverse Transcriptase Inhibitors. My master's thesis is done in the Laboratory of Molecular System Simulation under supervision of Professor Raimund Kazimierkiewicz. So firstly, I'd like to tell you more about the human immunodeficiency virus. So HIV is a lentivirus from retroviral family. The untreated HIV infection may develop into the AIDS syndrome, which annually kills thousands of people globally. So the genetic information of HIV virus is coded into the double-string RNA string, right? And when it comes to the life cycle of the virus, it infects the cells which present the CD4 receptors. So as you can see, after the fusion of the virus inside of the CD4 plus cells, we have the reverse transcription. So reverse transcription is a process in which the RNA, of this case viral RNA, is transcribed into double-string DNA, which then can be integrated into the host cell genome. That's why when it comes to the infection of the HIV virus, we can distinguish 4 major enzymes, which is the nucleases, proteases, integrases, and most important for my master's thesis, reverse transcriptase. So to treat HIV infection, we use highly active antiretroviral therapy, or HART. In this therapy, we use 2 or more classes of inhibitors of the enzymes which I have talked about previously, and including those inhibitors are the reverse transcriptase inhibitors. Those inhibitors are then subdivided into 2 subclasses: the nucleoside reverse transcriptase inhibitors, which are the analogs of the nucleosides existing in the human body, and the non-nucleoside reverse transcriptase inhibitors. So those inhibitors bind into the specific pocket of the reverse transcriptase, which is located around 10 Armstrong away from the polymerase active site. And on a picture, you can see them mark the amino acids which build this pocket. It's marked in magenta. It's around here. So in my master's thesis, I used 2 main approaches to generate and to score all of my molecules. So the first is genetic algorithm. This is the algorithm in which the population of the candidate compounds are scored, and then the best fitted particles are getting advanced, they proceed to the next generation. The least, or the worst particles generated are getting mutated in the next generation to hopefully become better. To score all of those molecules, I use molecular docking, which predicts the interaction between 2 molecules. In my case, I'll be using the protein-ligand interaction analysis, in which the protein is the reverse transcriptase, and then the ligand would be the molecules generated by the genetic algorithm. So when it comes to my master's thesis, I use the following programs: the autograph 4 and the quick VNA2 docking we use together. So the autograph 4 is used to basically manage the genetic algorithm, and then the quick VNA2 docking was used to score all of the generated molecules by the autograph 4. Also, to validate all of the results obtained by the quick VNA2 docking, I use the web service Doctor. And to visualize all of the structure and to edit all of the 3D structures in my all of that, I use the PyMol software. Then at the end, I also analyze my chosen molecules in Swiss ADME program for toxicological analysis and for bioavailability analysis. Also, I have built a whole database consisting of all of my generated molecules in Excel. So when it comes to my results, from around 10,000 newly generated molecules, I've chosen top 10 best fitted molecules, which structure you can see at this slide. Firstly, I'd like to compare the affinity to the reverse transcriptase. As you can see on this slide, on the right side of it, there are all of my 10 molecules in here. And the affinity of all of those molecules to the reverse transcriptase is basically the same, or at least they're similar to the inhibitors which are used today in the highly active retroviral therapy. Secondly, I also analyzed the bioavailability of all of those molecules. So in a slide, you can see the boiled egg chart, which analyzes the bioavailability of all of my molecules. In this slide, you can see that all of my 10 molecules are located into the white part of this chart, which means that those molecules will be highly absorbed by the gastrointestinal system. Then I also analyzed the potential toxicity and the drug likeliness of all of my molecules. So all of my 10 molecules, according to those filters which are listed in here, are drug-like, which means that they bear the similar properties, chemical and physiological properties, to the existing drugs. Also, what is important for all of my analysis is that no structural alerts were detected. It means that in all of those 10 molecules doesn't bear the same, at least doesn't bear the structural similarities to the existing carcinogens and to the existing toxins. That's a good thing. In summary, all of my chosen molecules have similar, or at least, or maybe even greater affinity to the reverse transcriptase when you compare that to the available inhibitors. Also, all of my chosen molecules are orally bioavailable, which is important because all of the HRT drugs are taken orally. And my chosen molecules are at least potentially least toxic, or at least they're non-toxic. So in big summary, all of my 10 molecules would be a great drug candidates, but that has to be researched further. Thank you so much for your attention, and I'll be waiting for your questions.
Okay, thank you very much. Any questions from the audience? Again, over there. I don't know. Two ladies first. Student. Okay.
So, yeah, do you hear me? So I have a question about this boiled egg, because you said that, yes, your molecules are all in this white part, but how about this yellow part? Because not every molecule is in the yellow part, so what does it mean?
So the main point with the boiled egg chart is, of course, the first, as I said, to analyze the absorption by the gastrointestinal tract, and this is in the white area. But also what you have in there, in this yellow area, is the blood-brain barrier permanence. So this, you can see from here that only not molecule number 8, they could permeate between the blood-brain barrier of our human beings. It is, for my master's thesis, it's not really that important, as the commercial, to be honest, used inhibitors are not they're not really like, you have inhibitors which permeate through the blood-brain barrier, but there are also some of them which don't, so it's not really that over deal breaker like other things which I mentioned in here. But I think that in most of future times, that all of the medicine should actually be coming here, because today it's much more, you have much more cases of the like neurodegenerative problems with in the HIV infected patients. So that's maybe a problem.
Okay, thank you.
Okay, I missed one small thing in your introduction. So why there is a need to design new drugs against reverse transcriptase if we have heart therapy?
So it's a very good question. The problem is that you have drug resistance to the heart therapy drugs used. Especially in the HIV-1 reaction, you have the drugs which are resistant to that, and that's why it's the same, to be honest, I think it's analogous situation to the bacteria, which also can develop the resistance to the antibiotics. And the same in here, we have to create a new classes or even a new generation, because today we are a second generation of those reverse transcriptase inhibitors, and creating a new ones could better the effects of this whole therapy for even the mutated.
Okay. Are there already programs that can predict like the resistance against drugs development?
To be honest,
that would be a good tool because you have a tool for everything like bioavailability, for toxicity, so also mutational rate would be interesting.
Maybe those tools do exist, but my problem is the lack of resources, right? All of my master's degree is done, to be honest, on my personal computer. And for that, maybe I don't know if such programs exist, but if so, they would use much more in computational power for me to be able to be honest, to have in here. So that's maybe the problem.
Okay. Thank you. Is there any, like a short question? Short one? No? Long one? Okay, we'll try to make it short.
So my understanding was that the basis for your new molecules were already existing drugs, right?
No.
Okay, so these are de novos.
No, those are completely done from scratch. To be honest, the program which is doing that, it uses the simulated chemical reaction to create all of those programs, but the source compounds, or to be honest, the baseline of all those compounds are the naphthalines, are the database of naphthalines. So it's based like benzene, etc.
So did you compare your molecules to the existing ones and how they basically in structural form compare, and that might tell you a bit about, for example, the resistance that you were already mentioning?
So the only thing, why do I compare all my 10 molecules to the existing inhibitors, is to just analyze the basically to tell everyone that all of my molecules are at least on a similar level to the affinity to the existing compound. So they're not worse in their job as the when you compare them to the existing inhibitors. So that's only my job. To be honest, I didn't really check about the resistance at all of the all those new drugs. So I didn't really find out, but yeah, that's a great point.
Thanks.
Good. We have to move on to the next speaker, so thank you very much once more. And the microphone. The next speaker. And we are changing the labs, and now we have a presentation from the Laboratory of Biologically Active Compounds, actually two. And the first presenter is going to be Antonina Nowak, which is telling us about green synthesis of silver nanoparticles using C. buckthorn.
Good morning, everyone. Today I would like to introduce you to my master thesis entitled Green Synthesis of Silver Nanoparticles using C. buckthorn, Hypophyra rhamnoides, a plant with antioxidant properties. I am working under supervision of Professor Aleksandra Królicka. As we all know, there is a global growing problem of antimicrobial resistance. Our current drugs don't work, and we are in desperate need to look for alternatives. Fortunately, there are some resolutions, such as silver nanoparticles, which are very small particles of silver, typically from 1 nanometer to 100 nanometers, and they are having antimicrobial activity. The best way to obtain them is to perform green synthesis, because it's environmentally friendly, since it avoids using toxic chemicals. And to form silver nanoparticles, silver ion must be reduced, and for that, plant extracts with antioxidant properties can be used, since they can donate electrons and act like reducing factor. The purpose of my research is to develop the best method for the green synthesis of silver nanoparticles using plant extract with high antioxidant properties to achieve the highest antibacterial efficiency, and also to check the interaction of the obtained silver nanoparticles and plant extract with high antioxidant properties in their antibacterial activity. I have searched through books and papers to collect database with plants having antioxidant properties. And here you can see 13 plant species that I have found, and collected to my research. To find out which of them has the best antioxidant activity, I performed DPPH method, which is standard technique used to measure antioxidant activity. And as you can see on the graph, the best antioxidant activity had Hypophila rhamnoides fruits, and that is why I then chose it to my further research. To develop the best method of synthesizing silver nanoparticles, I used 3 different methods: microwaves, water bath, and sonication. Moreover, I tried different conditions of reaction, such as different pH, different centrifugation parameters, different stabilizing agents, and I have also analyzed supernatant as well as precipitate. And as a result, I obtained 23 different samples, but since the time limitation, my next part of presentation will be only about 2 best methods and 2 best samples. So it turned out that the best methods were water bath and microwave. Those are 2 separate methods, one that uses microwave and one that uses water bath, but since it's their only difference, I decided to talk about them together. First, I prepared water extract from Hypophila rhamnoides fruits and adjusted its pH to 10. Then I mixed it with silver nitrate solution, water, and 3 sodium citrate as a stabilizing agent. And then I put the mixture into water bath or into microwave. After a given time, I had to wait until the mixture cooled down, and then I performed first centrifugation at low speed to get rid of the biggest aggregates, and I collected supernatant, and then I performed second centrifugation at high speed to separate the leftovers after reaction from my nanoparticles. And the last step was, of course, collecting precipitate. After I synthesized my nanoparticles, I tested their antimicrobial activity by performing microdilution method on 96 well plate, which enables to establish minimum inhibitory concentration and minimum bactericidal concentration. And as you can see, silver nanoparticles synthesized with water bath in case of Escherichia coli and Staphylococcus aureus works 5 to 6 times better than nanoparticles synthesized with microwave. And another thing that is worth noticing is that nanoparticles definitely worked better on gram-negative bacteria comparing to gram-positive bacteria. To establish size of my nanoparticles, I used dynamic light scattering method. And as you can see on the graphs, nanoparticles synthesized with microwave were size of 3 nanometers, while the ones synthesized with water bath were size of 12 nanometers. To check morphology of silver nanoparticles, I got images from transmission electron microscope, and we can see in all of those pictures, we can see different sizes. We can see sphere, rods, or triangles. We can see that they also differ in size, and that there is no very big aggregates, and that they are rather well spread out. The last step was checking the interaction between nanoparticles and extract from Hypophila rhamnoides leaves. The purpose of this test is to find out if it's possible to reduce dose of nanoparticles if we combine them with extract. And for that, I used tetrahydrofuran extract from Hypophila rhamnoides leaves because it had great antimicrobial activity. And to test this interaction, I performed checkboard titration method, which is a method that combines 2 agents in growing concentrations in a pattern that enables to establish their synergistic, additive, or antagonistic effect. And it turned out that in case of Escherichia coli, synergy occurred at 4 times lower concentration than it's needed when nanoparticles are alone in case of water bath method. And in case of microwave method, it was even 8 times lower concentration. Preliminary results also show that in case of Staphylococcus aureus, it is possible to reduce dose of nanoparticles even 8 times. Finally, conclusions. Among all investigated plants, Hypophila rhamnoides fruits
had the highest antioxidant activity. And by using extract from those fruits, I successfully synthesized silver nanoparticles. The method that used water bath and extract adjusted to pH 10 was the most effective. Gram-negative bacteria were about 25 times more sensitive to silver nanoparticles comparing to gram-positive bacteria. And last but not least, combining extract from Epiphyra rhamnoides leaves with silver nanoparticles enabled to reduce dose of nanoparticles 4 to 8 times. And in the future, we are planning to perform toxicity tests using model organisms Galeria melonella. Thank you for your attention.
Thank you very much. Are there any questions from the audience? Any question? Good.
Could you explain why silver nanoparticles can be toxic?
Yes. There are actually few ways that they can be toxic. First one would be that they are very small particles, so they can penetrate or enter cell membrane, disrupting it, and disrupt cell functions there. Another thing is that they can interact with proteins or enzymes within a cell and then create reactive oxygen species. Also, they are not always stable, so it is possible that they will release silver ions, which are very, very toxic.
It's okay? Good, okay. I think we have one question, no? Okay, good. Just get the info that we have to speed up a bit, so is there any other question? Short question? No? Okay, then thank you very much. Just take the microphone to the next speaker. And our next speaker from the same laboratory is going to be Anna Savik, which is presenting for us the diverse effect of Drosera zagzagia extracts on multi-drug resistance environmental strains of Escherichia coli.
Hello everyone, my name is Anna Savik and I'm glad to present you the results of my master thesis project that was dedicated to examining the diverse effects of Drosera zigzagia extracts on multi-drug resistant environmental strains of Escherichia coli. Antibiotics serve as a main way of fighting the microbial infections. They are also used widely in animal husbandries and agriculture, but unfortunately they are not completely metabolized and could be released into the water environment via wastewaters, where they can cause the development of resistance to antibiotics, for example in Escherichia coli. Also, such mechanism as horizontal gene transfer can also contribute to the spread of resistant genes. Moreover, from hospital wastewater, we can have clinical isolates that can be resistant to some antibiotics and also be pathogenic for us. So they survive sewage wastewater systems and also can be found in water and cause risk for potential water users. So this infection could be extremely difficult to treat. That's why we need an alternative solutions, and we can use carnivorous plants extracts for that. The good example of such plant is Drosera zigzagia that naturally grows in Western Australia, and its extracts contain, among other properties, a lot of bactericidal effect. By cultivating those plants in vitro, we can create the environment where we can propagate rare plants and accelerate their growth. Also, those plants are pathogen-free, and what is the most important, we can enhance the production of secondary metabolites by introducing a stress stimulus. So, aims of my study were following: investigation of the biodiversity of Escherichia coli strains in the water reservoirs of the Gdansk Bay.
optimization of secondary metabolite extraction from the zigzag using various extraction methods, and assessment of the elicitation impact on the activity of Drosera zigzagia extracts against reference strains Staphylococcus aureus and reference and environmental strains of Escherichia coli. Bacteria samples were isolated from the coast of the Gdansk Bay, and to analyze the biodiversity, they were collected from different dates from various locations using different isolation methods. We have samples from seawater, fresh water, and wet sand. And what is very important, that those strains were not isolated for drug resistance at the first place. So after I obtained the genome profiles, performing repetitive element PCR with box primers, I assigned them to different phylogroups doing quadriplex PCR and determined the resistance to 16 antibiotics from 7 different groups. Drosera zigzagia was cultivated in vitro, firstly on solid medium, then on liquid medium, with 2 stress factors, shaking and cultivating plants on different medias with various nutrient composition and pH level. Next, extracts, secondary metabolites were extracted using 3 different methods: water extraction, salt out assisted liquid extraction, and SOX-less extraction. And at the end, the antimicrobial properties were tested by establishing minimal inhibitory and bactericidal concentrations. Moving to results section, the heat map illustrates the resistance of E. coli strains to 16 antibiotics, and blue areas illustrates the small inhibition zones. That means that we have bacteria that are resistant, for example, to ampicillin. On the other hand, red areas shows that we also have susceptible strains, for example, to carbapenem group of antibiotics. But let's add some numbers to this graph. From all isolates, I found 20% that are resistant to at least 1 group of antibiotics, with ampicillin being the most frequent one. What is also interesting that I found the resistance to almost all antibiotics from cephalosporin group. Those resistant strains were mostly isolated from Gdynia, but as you can see, they were mostly resistant only to 1 antibiotic, while in Gdansk, these samples were more diverse, with a resistance to higher amount of various antibiotics. Division bacteria into different phylogroups answers the question, how many of them could be potentially pathogenic? And I found 20% of bacteria that contribute to B2 and D group that are mainly associated with pathogenicity. And in this 20%, there are resistant ones. And what is very important, that multi-drug resistant strains are a part of B2 group. And finally, the antimicrobial properties were tested. And on the left graph, you can see the differences between extraction methods, with salting out extraction being the best one, as lowest concentrations of the extracts were needed to obtain MBC. And here you can see that elicitation wasn't affected the E. coli strains, but for S. aureus, the extracts from plants that were cultivated on 1/4 Murashey and Scoop medium were working better. That's why I decided to choose these particular extracts to check how it works on environmental strains. And as you can see, it has an antimicrobial effect on resistant environmental strains. The concentration needed to obtain MBC were always a little bit higher, but generally speaking, there were no connection between resistance to antibiotics and reaction to the plant extracts. In conclusion, 20% of all the strains were resistant at least to 1 group of antibiotics. 30% of them belonged to phylogroups B and 2 and D. The highest number of drug-resistant antibiotics were found in Gdynia. Elicitation of Drosera zigzagia by culturing on different media affected only against S. aureus, and saltinol assisted liquid extraction was the most efficient one. And last but not the least,
The zigzag extract proved to be efficient against both multi-drug resistant and antibiotic susceptible environmental strains. I want to thank the Laboratory of Biological Active Compounds and especially to my supervisor Marta Potrikus and Professors Aleksandra Kurlicka and Eva Wojkowska for their support and help throughout this time. Thank you for attention.
Thank you very much. Questions from the audience? Someone has a question? Okay.
I would like to ask if there are any specific components of these extracts that are responsible for their bactericidal properties.
Yes, actually, I'm also testing this, and literature says that there are some compounds like naphthoquinones, for example, that could have this effect, could be responsible for such properties of the extracts. And for example, it's plumbagene. And I'm performing thin layer chromatography to establish if it's present on my extracts or not. But I still need some time to perform more precise analysis on that topic.
Okay, thank you.
Thank you very much. Any more quick questions, short one? No, I think that we can also leave it with that. Thank you very much once more. And you can also just show the microphone. The microphone. Okay, good. Okay, and our next speaker is going to be from the Laboratory of Plant Biochemistry. It's going to be Patrycja Gruszewska, which is telling us about the effect of knockout of gene encoding for phospholipid sterol acetyltransferase on physiology and development of Arabidopsis thaliana grown under abiotic stress conditions. You ready? Good.
Good afternoon. The title of my master project is Effect of Knockout of Gene Encoding for Phospholipid Sterol Acetyltransferase on Physiology and Development of Arabidopsis thaliana grown under abiotic stress conditions. I'm working in Laboratory of Plant Biochemistry under supervision of Katarzyna Jasińska-Gazarkiewicz. So in Arabidopsis thaliana, there are two enzymes which are responsible for sterol ester synthesis. First one is PSAT, which transfer acyl group from phospholipid to sterol. Second one is ASAT, which transfer acyl group from acyl coenzyme A to sterol. Free sterol are membrane components, while sterols conjugated with fatty acids, so sterol esters, are stored in lipid droplets in cytoplasm. So PSAT, by its activity, regulate the amount of free sterols in membrane. Changing of components of membrane is called membrane remodeling, while negative impact of environmental factor is actually abiotic stress. So in my study, I'm trying to answer the question, is phospholipid sterol acetyltransferase as an enzyme which are involved in membrane remodeling is also involved in plant response to abiotic stress. To answer this question, I ordered Arabidopsis thaliana mutants which have tDNA insertion in gene encoding for PSAT. I have two lines of mutants which differ in the position of tDNA fragment within the gene. These are the names of mutants lines, but to simplify, I named them PSAT-KO91 and PSAT-KO89. And first what I did, I designed primers, I isolated DNA, and I amplified gene fragment to prove tDNA insertion. Then I started cultivating plants in vitro and in vivo in standard, salty, and low temperature conditions. I performed morphological observation. Then I collected material for lipid extraction. I methylated lipids to be able to perform gas chromatography analysis. I isolated microsmall fractions. I determined the protein contact. I did enzymatic reactions. Then I separated a product of this reaction by thin layer chromatography. And I determined concentration of product. Morphological observation demonstrate that there is no differences between wild type and mutants in standard conditions. In salt stress, the differences are only in fresh weight of roset, whereas in low temperature conditions, differences are in all measured parameters. What's interesting, I found that these differences under low temperature stress appears only when stress conditions are introduced before 0 days after flowering. Although PSAT substrate and product are lipids, there is no changes in total lipid content in rosettes, roots, and seeds in all tested conditions. But if you look at the representation of particular fatty acids, there are some changes. In rosettes in standard conditions, PSAT KO89 has less 18,1 fatty acid. There is no differences in salt stress, whereas in low temperature stress, PSAT KO91 has less 16,0 fatty acid and more 16,3. In roots, there are no changes in standard conditions and also in salt stress. And in salt stress, there are large standard deviations. This is because salt stress greatly inhibits root growth, making precise measurement very difficult. In seeds, in standard conditions, mutants accumulate less 16,0 fatty acid and more 18,1. Also, PSAT KO89 has more 18,2 fatty acid, whereas PSAT KO91 less 21. Again, in salt stress, there are no differences. In low temperature, there are lots of differences. And what's interesting, the mutants have less saturated fatty acid and more unsaturated fatty acids. I also tested PSAT and ASAT enzymatic activity in wild-type Arabidopsis thaliana, and I found that activity of both enzymes is higher in microsomal fractions isolated from plants growing under abiotic stress conditions, and also that magnesium and calcium ions stimulate the activity of PSAT and ASAT. So based on my result, I draw a conclusion that PSAT is involved in plant response to low temperature stress, and that knockout of gene encoding for PSAT does not affect the total lipid content, but affects the composition of particular fatty acids. And also that abiotic stress affects PSAT and ASAT activity, and this activity is stimulated by magnesium and calcium ions. Thank you for your attention.
Okay, good. Thank you very much. Any questions from the audience from this presentation? Anyone has something? Because I also didn't really have anything to ask about. Can you go back to one slide? Because I see that in the lower graph for the MG2 and the CA2 in the enzymatic activity, the middle bar is quite tall compared to the other two. Can you do you have any idea where this comes from, or any kind of, maybe insight why this is?
Actually, we don't know, but but maybe these ions are like important for the structure of enzyme. Maybe it's incorporated in the structure of this enzyme, but actually we don't know. This is like my idea. It's guessing.
Okay, cool. Any other question over there? One. We have to go all the way around.
Okay, thank you. Very nice presentation. In one place, okay. I will try. I will do my best. Okay. Maybe we can try without that.
Yeah. Is it okay without?
Is it okay?
Yeah.
So my question is about the phenotypic differences that you show
I did not test it how the lipid content look like when the plant is introduced to stress condition after 0 DAF. I only like have this morphological observation. Mm-hmm. That these morphological differences appears only when the plant is introduced to stress conditions before 0 days after flowering, but I didn't test the lipid content. In these flowers which were introduced to stress conditions after 0 day after flowering.
Okay. Good. Okay, I think that's enough questions, and thank you very much once more. And our next speaker is from the Laboratory of Plant Microbiology. It's going to be Natalia Gdansk. I hope I don't mispronounce the name. And she's presenting for us the effect of colonization of Arabidopsis thaliana tissue by the beneficial strain Pseudomonas donghensis P482 on plant growth and condition.
Okay, so the title of my diploma thesis is Effect of Colonization of Arabidopsis thaliana tissues by the beneficial strain Pseudomonas donghensis P482 on plant growth and condition, and I did my research at Laboratory of Plant Microbiology under supervision of Magdalena Rajewska. The main object of my research is Pseudomonas donghensis P482, and it is a gram-negative aerobic bacterium isolated from the rhizosphere of a tomato plant in Gdynia. It exhibits antimicrobial activity against phytopathogens, including bacteria and fungi. It is also an efficient root colonizer of course, tomato, but also potato and maize. And it is relatively new bacterium, so it's still under investigation. Okay, so root colonization is the most important process that facilitates the interaction of plants with bacteria. And it is possible mainly through biofilm formation by these bacteria. And biofilm formation is tightly controlled in bacterial cells by various signaling pathways. And in this study, I focused specifically on CDGMP and two-component system GAC-SGAC-A. And CDGMP is a key signaling molecule in bacteria that controls the transition from motile lifestyle to biofilm one. And two-component system GAC-SGAC-A is a key regulatory pathway in Pseudomonas that controls the expression of genes involved in biofilm formation, but also secondary metabolites production and quorum sensing. So I investigated whether P482 and specific mutants are capable of colonizing roots of Arabidopsis thaliana and what impact they have on its growth. So these mutants I mentioned include 4 mutants in CDGMP metabolism and 1 mutant defective in GAC-A gene associated with two-component system GAC-SGAC-A. Aims of the study were to analyze the effect of colonization of Arabidopsis thaliana tissues by Pseudomonas donghensis P482 on plant growth and optimization of methodologies for analysis of this interaction. The methods I used included 3 experimental setups. So for root system analysis, I used square plates with medium modification for bacterial and plant growth, and then I measure 3 parameters, so length of the main roots and length and number of lateral roots. For mass analysis, I cultured inoculated seedlings in flasks with shaking, and then I measured wet and dry mass of shoot and roots. And for colonization analysis, I used GFP tag strains, cultured them in gnotobiotic conditions, and then visualized roots under scanning confocal microscope. So moving to the results, so first influence of P482 and its mutants on the root system growth. And the first graph refers to average length of the main root of Arabidopsis after treatment with a given bacterial strain, as well as the controls that received no treatment. And as we can observe, that there are no significant differences between the treated and untreated groups. And the next 2 graphs show the root system architecture, so number and length of lateral roots. And among all strain tested, there is a slight increase in these parameters for P482 wild type, so this pink one, comparing to the control. And additionally, this mutant 4501, so this green one associated with CDGMP, exhibits similar values to the wild type. Next experiment aims to assess the impact of P482 and its mutants on mass increase of shoot and roots of Arabidopsis. And the first two graphs show the wet and dry weight of the shoot. And as observed, mass of plants treated with the wild type strain is lower comparing to the control. Mutants associated with CDGMP exhibit similar values to the wild type. And GAC-A mutant, however, leads to a significant increase in shoot mass. In case of the roots, we observe similar trend. So this wild type strain leads to a reduction in roots mass comparing to the control, and the GAC-A mutants result in a significant increase of the mass of the roots. And in the last experiment with scanning confocal microscope, the goal was to visualize colonization of Arabidopsis roots. And we can see, it is visible here that all mutants efficiently colonize the roots of Arabidopsis. So these first two associated with CDGMP, but we can see slightly different pattern of this colonization. So this first two associated with CDGMP form more smaller dispersed microcolonies. The next two form larger bead-like microcolonies, and the GAC-A mutant, however, form more flatter layers on the root surface. And to conclude, so mutations in genes associated with CDGMP and the GAC-S-GAC-A system do not significantly impact root system of Arabidopsis thaliana based on these results. Secondly, mutations in genes associated with CDGMP result in a reduction of both shoot and root mass, and conversely, the GAC-A mutant leads to an increase in shoot and root mass in liquid medium. And finally, P482 and its mutants associated with CDGMP, as well as the GAC-A mutant, are able to colonize the roots of Arabidopsis. Thank you.
Okay, thank you very much for this presentation. Is there a question from the audience regarding that presentation? Yes, I can. Okay. Road? I will just do without. Okay.
So my question is to the negative control you showed on one of the pictures, because you have two types of, yeah, like this one, or next one. Next one, please. So this is just the Arabidopsis wild-type plants without bacteria.
This like all the wild-type plants, but what differ is like the different strains of bacteria.
Yes, I understand. So you have wild-type plants on the
Yes,
one side, on the
without
other
bacteria.
side.
So this negative control refers to plants without the treatment, so without bacteria.
That's what I mean. So if you look at the slide, you have like plants without colonization, then you colonize them with two types of mutants, let's say, right? The JCA mutant, and the other 5 mutants belong to GMP metabolism.
Yes.
Yes. Yes, so that's my question, because if you look at the, so in your conclusion, correctly, you said that if you add the mutant JCA, you have increase in biomass.
Yes.
Yes. But if you look at the other group of mutants, like GMP metabolism, and you compare it into negative control.
No, I did compare it with a wild type of these bacteria. So.
Okay, but this is the growth of the plants here.
Yes.
Yes. Okay. We can discuss it later. Thank you.
Okay, yeah. Any other questions from the audience? No. Okay, so then thank you very much once more. And now we're going to have 3 presentations from the Laboratory of Plant Protection and Biotechnology, where the first speaker is going to be Katarzyna Kaczerska, which is presenting the analysis of extracellular vesicles secreted by bacteria of the Dickia genus. And I think she's already there. Okay.
Hello everyone, my name is Katarzyna Kaczerska, and today I want to present some of the results I've obtained during my master project called Analysis of Extracellular Vesicles Secreted by Bacteria of the Dickia genus. I've conducted it under the supervision of Professor Małgorzata Valeron with the assistance of Dr. Joanna Jońca in Laboratory of Plant Protection and Biotechnology. Bacteria of the genus Dickia are gram-negative members of family of Pectobacteriaceae, and due to the presence of 2 membranes in the cell envelope, they contain different types of secretion systems through which they can secrete, for example, pectinolytic enzymes. They can cause wilting and diseases such as a blackleg and soft rot. Apart from the plants, they can survive in soil, water, and insects, which facilitates their transmission, and currently they are widespread globally. Bacteria, like other organisms, can produce extracellular vesicles, which are lipid bilayer particles that take part in interactions between organisms or in eliminating unwanted substances. By some researchers, they are referred as type 0 secretion system, and in 2021, some members from our team described vesicles produced by other member of Pectobacteriaceae family, Pectobacterium, noting their potential role in the interactions with plants and in virulence. However, vesicles secreted by Dickia species have not yet been described. For this reason, my aim of the study was to isolate, characterize, and compare vesicles secreted by Dickia gdanskii and Dickia solani. I also wanted to determine the influence of lack of various secretion systems and of membrane disruption on the content and number of secreted vesicles. And at the end, I wanted to establish the effect of direct interactions between Dickia and Arabidopsis in vitro. During this study, I used Dickia gdanskii 3937 strain along with a mutant that lack genes that affect activity of type 2, type 3, and type 6 secretion system. And for Dickia solani, I use IPO2222 strain with a mutant that lacks DSBA gene, which causes membrane instability and disruption. After bacteria cultivation, I performed isolation of the vesicles, which were later visualized under the transmission electron microscopy and by nanoparticle tracking analysis. Proteomic analysis told us about the content of the vesicles, and based on those results, we tried to predict possible interactions between proteins of those vesicles and Arabidopsis thaliana. And at the end, I performed the study of interactions between bacteria and Arabidopsis thaliana in vitro. Moving to the results, we determined that all tested Dickia strains can produce vesicles. Here are the pictures obtained from transmission electron microscopy and a video of the vesicles from nanoparticle tracking analysis. There is no significant size difference between strains. However, Dickia gdanskii produced more vesicles than Dickia solani, and additionally, mutants with inactive type 2 secretion system and with unstable membranes increased the vesicles production, while mutants with inactive type 3 and type 6 secretion system decreased the production. After proteomic analysis, we determined that both Dickia gdanskii and Dickia solani produced similar number of the proteins, with 28 of them common for both species, which were mainly outer membrane proteins. However, we found their protease precursors PRTB and PRTC, which are virulence factors. After further analysis, we determined that Dickia gdanskii produced more unique, secretes more unique more proteins in their vesicles with protease precursors PRTA and with virulence protein DIRK. When I compared the results from Dickia gdanskii 3937 strain to the results obtained from different mutants of secretion system systems, we determined that there is a drastic change in the protein's composition with 9 proteins common for all species. They were memory proteins, chaperone proteins, translation proteins, and precursors of proteases A and C, which are once again virulence factors. When I analyzed the proteins that are exclusive for vesicles secreted by type 3 secretion system mutant, I identified PALD, which is a pectatolyase that can cause pectin degradation in the cell wall of the plant. Mass spectrometry results were further analyzed with ARA pathogen software, so in silico analysis, to determine the possible interactions between proteins that are secreted in the vesicles and those of Arabidopsis thaliana's plant. And despite those big differences in the composition of the vesicles, I determined that there are 270, that those proteins can participate in the interactions with with almost the same set of the proteins that belong to Arabidopsis thaliana, and those proteins are taking part at many biological processes during life of Arabidopsis. So therefore, more studies are needed to confirm this. And at the end, I performed the study in vitro when I tried to establish the effect of interactions between Dicea dadanti and Arabidopsis. And those bacteria could produce accessory vesicles to the environment. And after these interactions, we noticed that there is a significant change in the roots length. And after imagination under the light microscopy, we noticed that that morphology of the roots is changed with increased number of lateral roots and root hair. And this is the end of my presentations. Here are the most important points from it. All tested strains of Dicea solani and Dicea dadanti can produce vesicles of the similar size. However, Dicea dadanti produced more vesicles compared to Dicea solani. Mutations have big influence on the amount of secreted vesicles, as well as on the number and the composition of the proteins in those vesicles. However, all vesicles that were analyzed contain virulence factors, and during interactions, all studied Dicea strains reduce morphology, change morphology of the Arabidopsis roots and reduce their length. Thank you very much.
Okay, thank you very much. Any questions from the audience from this presentation?
Okay, so I have a question. How did you isolate extracellular vesicles?
After the bacteria cultivation in the media in the mineral media with pectins, maybe I'll go to the slide. After the cultivation of the bacteria, I centrifuged them to discard all the bacteria and all unwanted substances, and to be sure that those vesicles aren't contained any bacteria. I performed filtrations through 2 membranes to discard everything. And after this, I performed the ultra-centrifugation, and this is how I obtained them.
Okay, thank you. Any other questions? No. Okay, thank you very much. Then, and our next speaker is going to be Agata Szczepanska from the same laboratory. She's going to present for us the pro-apoptotic effects of piperlongumine towards tamoxifen in estrogen receptor positive breast cancer cells. Yeah.
So good afternoon. My name is Agata Szczepanska, and today I'm going to present my master thesis, and the title is Pro-apoptotic Effect of piperlongumine toward tamoxifen in estrogen receptor positive breast cancer cells. I'm working under... Okay, so maybe...
So, hello everyone, my name is Agata Szczepańska and today I'm going to present my master thesis, and the title is Pro-apoptotic Effects of Piperlongumine toward tamoxifen in estrogen receptor positive breast cancer cells. I'm working under the supervision of Dr. Anna Kawiak in Laboratory of Plant Protection and Biotechnology. And at the beginning, I would like to briefly talk about subgroups of breast cancers. As you can see, we can distinguish 5 groups, and 3 of them, luminal A, normal-like, luminal B, contain hormone receptors. And as a target therapy, tamoxifen is used. But unfortunately, more and more breast cancer cells becoming resistant to tamoxifen. So there is a need to explore new compounds which can be helpful to modulate the activity of tamoxifen. And aims of my study: first, I want to define the pro-apoptotic effect of piperlongumine in estrogen receptor positive breast cancer cells. Then to specify if piperlongumine induce apoptosis through the JNK signaling pathway, and also to determine if piperlongumine can increase the induction of apoptosis in estrogen receptor breast cancer cells by tamoxifen. But what exactly is piperlongumine? Piperlongumine is alkaloid, which is naturally found in long pepper, and this bioactive compound possesses many therapeutic properties. And also, piperlongumine has demonstrated ability to induce apoptosis in cancer cells while having less impact in normal cells. And perhaps in breast cancer cells, piperlongumine induce apoptosis through the JNK signaling pathway, and this pathway is crucial in controlling cell proliferation and also cell response to stress. So let's move on to methodology. To determine the cell viability, I performed MTT test and also colony-forming assay. To see the profile of cell population, I conducted flow cytometry. And to check if piperlongumine induce apoptosis through JNK signaling pathway, I performed alpha screen assay. And this is the quantitative method. And as you can see, when analyte is present, it triggers the formation of sandwich complex. So then when donor molecule is excited, it triggers, the acceptor molecule can emit the light at 650 nanometers. So now let's focus on result. I worked on 2 estrogen receptor breast cancer cell lines, MCF7 and also T47D cell line. And as you can see, these graphs show how the cell viability was affected by a different concentration of piperlongumine and tamoxifen after 48 hours of incubation and after 72 hours of incubation. And as we can see, the optimal time for piperlongumine, it was 48 hours, and in case of tamoxifen, it was 72 hours. And more sensitive to tamoxifen and piperlongumine was T47D cell line. And then I performed flow cytometry, and as a control, cells were incubated with only medium. And as we can see, in control, there was around 11% of apoptotic cells. After the treatment with 5 micromoles of piperlongumine, there was 22% of apoptotic cells. And after treatment with 10 micromoles of piperlongumine, it was around 45%. And to check if piperlongumine induced apoptosis through JNK signaling pathway, I pre-incubated cells with JNK inhibitor. So, and after the pre-incubation, I add piperlongumine and cells were incubated for 48 hours. And as we can see, after the treatment with only piperlongumine, there was around 60% of apoptotic cells. And after adding the inhibitor, the percentage of apoptotic cells reduced to 40%. And to directly confirm that piperlongumine induced apoptosis through this JNK signaling pathway, I performed alpha screen assay. And this graph shows the phosphorylated JNK levels normalized to total JNK level. And as you can see, as the concentration of piperlongumine increase, the concentration of the levels of phosphorylated JNK also rise. And then to analyze if piperlongumine can induce the pro-apoptotic activity of tamoxifen in estrogen receptor positive breast cancer cells. First, I have to find the optimal concentration of both compounds of tamoxifen and piperlongumine. So I treated cells with a different concentration, and then I stained them with crystal violet. And the optimal concentration of both compounds, it was 2.5 micromoles. So then I treated cells with only this concentration, and as you can see, in case of piperlongumine, there was around 26% of apoptotic cells. In case of tamoxifen, it was around 17%. And after combining piperlongumine with tamoxifen, it was around 50% of apoptotic cells. So to conclude, piperlongumine induces the apoptosis in estrogen receptor positive breast cancer cells, and the induction is through the JNK signaling pathway. And also piperlongumine increases the pro-apoptotic activity of tamoxifen in estrogen receptor breast cancer cells. Thank you.
Thank you very much. Are there any questions from the audience for her? Anyone? Something? Because I have anything as well. The two cell lines you used, why you used exactly these two cell lines?
Because, like I said this before, because in this case it was only like estrogen receptor positive breast cancer cells, and in case of these two cell lines, there is a presence of estrogen receptors. So that was the main focus in this study.
Okay. Other question?
I think so, yes. So it's possible, but on this study, I only focus on this cell line, but maybe in the future, I think there's a need to explore also like normal cells.
Okay, good. Any other questions? No. Okay, thank you very much. And then we are coming to the last speaker of this session, which is going to be Weronika Szkoda. She's going to give a presentation about the eradication of bacteria associated with acne vulgaris from ex vivo porcine skin by direct cold atmospheric plasma jet type DBD.
So good morning, everyone. Today, my name is Weronika Szkoda, and today I would like to present to you my master thesis project, which titled is Eradication of Bacteria Associated with Acne vulgaris from ex vivo porcine skin by direct cold atmospheric plasma jet type dielectric barrier discharge, shortly DBD. So to begin with, acne vulgaris is a chronic inflammatory and recurrent skin condition which affects approximately 80% of adolescents in aged between 11 to 30. It is 8th most frequently appearing disease globally, and one of the main 4 pathogeneses is abnormal colonization of follicles by Cutibacterium acne. The most current common way of treatment of this disease is to prescribe antibiotics to patients. And as we well know, currently we are facing the problem with rising antibiotic resistance. That's why it is so important to develop a new therapeutical approach. Here comes plasma, which is the 4th state of matter. It is simply explaining just ionized gas. Cold atmospheric pressure plasma generates free radicals, free reactive nitrogen species and reactive oxygen species, which interacts with biological cells and tissues. In plasma stream, we can find these radicals which interacts with cytoplasm of bacteria cell. Also, they lead to damage in DNA and RNA, and they have impact on cell membrane, cell wall, and so on. The aim of my project is to examine the antimicrobial effects of the electric barrier discharge, helium plasma, against selected bacteria associated with the symptoms of acne vulgaris. Here are my bacterial strains that I have selected. These bacterial strains are most commonly appearing in the tissue samples collected from patients with this disease. And first, what I did was the MALDI-TOF MS identification of the selected bacteria strains, only to confirm that I'm working with these particular strains that I wanted to test. As for a plasma generator, it was the electric barrier discharged, and the gas that I was using was helium. And thanks for atmospheric conditions, this type of plasma is cheaper to obtain than in vacuum. My experiment can be divided into two different methods. The first one was flat surface model, and the second one was porcine skin model. As for flat surface model, it was necessary to confirm that plasma stream eradicates my selected bacteria from a flat surface model, and then I could move to the second step, which was porcine skin model. And firstly, it was necessary to obtain sterile porcine skin, then to do artificial inoculation on this skin with my selected strains. And after the incubation, I was treating the bacteria directly on the surface of the skin with the DBD plasma. And after the exposure time, I was collecting them with the cotton bud, do some serial dilutions, and streaking them to the TSA medium plate. Here you have a flat surface model, how it was looking like in the real life, and here is the porcine skin model. So moving to results. As for efficacy of eradication from flat surface model, here are the representation for two different strains of Staphylococcus aureus. And as you can see, after 30 seconds, we have significantly reduction of number of colonies in comparison to the control. Here is the total percentage of reduction for a flat surface model. And for 8 strains, I have obtained more than 99% of reduction after 30 seconds of exposure to DBD plasma. Only for one strain, which was Staphylococcus aureus 209P, it was necessary to prolong the exposure time to 1 minute to obtain 96% of eradication. Here are the same result, but presented in the logarithmic scale. Here is the representation for 30 seconds, and here is the representation for 1 minute. And the value 0 means that there was total reduction. Moving to porcine skin. So here I tested the exposure time of 30 and 1 minute, and 30 seconds, obviously, and 1 minute. And here are results for Cutibacterium acne. And as you can see, there is almost a complete reduction after 30 seconds of treatment in comparison to control. But in contrary, for Staphylococcus epidermidis, we can see the reduction, but it's not a complete one. And this way of streaking allows me to establish accountable dilutions for the next experiment, which was, the difference is that the exposure time was set to 2 minutes. And as you can see, for all tested dilutions, we can see a significant reduction in number of bacterial colonies growing on the TSA medium plate. And this is the final result, which is the exposure time of 4 minutes for my selected bacterial strains. And here is the logarithmic reduction, here is the percentage of reduction. And for Cutibacterium acne, which is the most important bacteria strain for my studies, we can see the total reduction of 30% in average, which is definitely lower than from flat surface model. but for all strains, I received reduction. So in general, obtaining porcine skin, the sterilization of the porcine skin is really challenging. And cultivation of the bacteria is also really hard on the surface of the skin. Also, every single piece of skin varies, differs between the repetitions. So there are some challenges, but in general, DBD plasma is significantly reducing the number of strains, bacterial strains, and it can be a good addition to currently used therapeutics methods. And maybe in the future we are going to develop a mobile DBD plasma jet, which are going to be found in beauty salons. So thank you for your attention.
Thank you very much. And are there any questions from the audience?
You mentioned that the sterilization of this skin model, right, is challenging.
Yeah.
Can you tell anything more about how this process looks like, or how did you check if this skin is really sterile?
Okay, so firstly, after obtaining the skin, it was necessary to reduce the meat and the rest of the tissue from the skin. And then I was washing it with 70% of ethanol or 70% of isopropanol, then with previously sterilized water, and I did it 2 times. Then I exposure the skin for 20 minutes to UV lights from each side, and then I put it into -80 degrees to freeze. And this process I had to repeat 5 times to obtain sterilized porcine skin. So that's why it was pretty challenging.
Thank you.
Okay, any more questions from the audience? Doesn't look like it. Okay, thank you very much. That concludes the second session. I would like to thank all of the presenters and all the attendees in the audience. We're now going to have a coffee break until 13:30, so for 40 minutes. Afterwards, there's going to be the poster session outside in the lobby. That's going to take 1 hour until 14:30, and then 2:30, we are going to meet back here for the third session. Thank you. Welcome everybody back. I hope you had a nice break, and I hope there was a nice discussion about the posters. We're now going to have 5 presentations, and the first speaker is going to be from the Laboratory of Photobiology and Molecular Diagnostics. I would like to welcome Julia Sadowska with the title The Influence of a Photodynamic Method and the Novel Venetin-1 Nanoparticle on a Lung Cancer Cell Line.
Hello everyone, my name is Julia Sadowska, and I will present you my master thesis entitled The Influence of a Photodynamic Method and a Novel Venetin-1 Nanoparticle on a Lung Cancer Cells. And I did my research in the Laboratory of Photobiology and Molecular Diagnostics under the supervision of Dr. Magda Rybicka-Misiejko. And at the beginning, a few words about lung cancer. So this is the most common cause of cancer deaths worldwide. And most of the patients have a histological subtype known as non-small cell lung carcinoma, and I was working with that type of line A549. And Venetin-1 is a protein polysaccharide complex obtained from the celomic of Dendrobena veneta earthworms with proven anti-tumor activity against lung cancer cells. And recent studies on the proteome response of the cells to Venetin-1 showed no significant effects on control cells, while lung cancer cells strongly responded. And it was observed increased levels of proteins related to the activity of the mitochondrion and the endoplasmic reticulum, and proteins associated with glycolysis and gluconeogenesis were decreased. And interestingly, Venetin-1 caused a decrease in levels of protein biomarkers associated with susceptibility to cancer radiotherapy. And aims of my study was to examine the effects of Venetin-1 and red light on viability, cell cycle, cell death, and proteome of lung cancer cells. And now we can move to materials and methods. So I was working with a cell line that I already mentioned about, A549, which is non-small cell lung carcinoma. But I also had a control cell line, BS2B, which is normal bronchial epithelium. And at first I was seeding the cells and allow them to adhere. The next day I was changing the medium with a fresh one, a regular medium or a medium supplemented with Venetin-1. And after medium replacement, I was irradiating the cells with different doses of red light for 30 minutes, or I was keeping controls in the dark. And as a result, I had 4 variants of cells. And after incubation, I was preparing the cells for subsequent experiments. And it was examination of cells' morphological changes, analysis of cell cycle and apoptosis with flow cytometry, and analysis of proteome with mass spectrometry. And now I will show you my results. So at the beginning, I wanted to check the influence of red light on cell morphology. And here you can see pictures taken before irradiation, and here 72 hours after irradiation. But there is no differences in cell morphology in both cell lines. And then I wanted to examine the influence of only red light on cells, apoptosis, and cell cycle. And as you can see, different doses of red light didn't cause the apoptosis of lung cancer cells and didn't alter their distribution in the cell cycle. And the same is for control cells BS2B. Here is the influence of Venetin-1 and red light on lung cancer cells distribution in the cell cycle. And based on these graphs, we can say that treatment of the cells with venetin-1 and irradiation caused their arrest in G1 phase, and there was also less of those cells in G2M phase, where cells are prepared for division. But for me, the most important comparison was between cells treated with venetin and irradiated cells treated with venetin. And there is one difference between those groups in a sub-G1 phase, in the case of 75% power of red light. And that's why I chose to use this dose of light in next analysis. And here again, you can see this difference in sub-G1 phase. What indicated that venetin-1 in combination with red light increased the number of apoptotic cells compared to the treatment only with venetin-1. And I also performed the same analysis for control cells, but there is no differences here. And those are results from analysis of apoptosis of lung cancer cells. And here is the percentage of viable cells in early apoptosis, late apoptosis, and necrosis. And statistical analysis showed that venetin-1 with red light decreased the number of live cells compared to the treatment only with venetin-1. And this is the graphic representation of co-enrichment of the lung cancer cells treated with venetin. And marked proteins were downregulated and associated with cancer susceptibility to radiotherapy. But unfortunately, red light didn't enhance the proteome response of those cells to venetin-1, and SWAT mass spectra analysis allowed for identification of 706 proteins, but none of them exhibited any differences. And to sum up, red light doesn't affect lung cancer cells and control cells morphology, apoptosis, and cell cycle. And the combination of red light and venetin-1 on lung cancer cells increases the number of necrotic cells and decreases the number of viable cells compared to the treatment only with venetin-1. And control cells are not affected. And red light enhances the anti-tumor potential of venetin-1. And at the end, I would like to thank habilitated Dr. Marta Fiołka from Maria Curie Skłodowska University for providing me venetin-1 for all of the experiments. And I would like to thank the whole team of the Laboratory of Photobiology and Molecular Diagnostics, especially to my promoter for a huge support during this year. And thank you for your attention.
Okay, thank you very much. Any questions from the audience?
So at the beginning, you have to obtain the coelomic fluid. It's done by multi-electrode stimulation of Dendrobena veneta. Then you have to separate it from coelomocytes, filtrate, and the most important step is to heat this coelomic fluid to eliminate cytotoxicity. And then you lyophilizate it, and this is how you get venetin-1.
Okay, there's one more question. I just have to go around.
So you showed no proteomic changes from the red light.
Yes.
So what's your hypothesis?
What's the hypothesis behind this increased necrosis caused by the red light? It's not a change in the proteome, so no expression, This is a difficult question, and honestly, I don't know yet exactly why it happened. I don't know.
Okay, maybe that's something to look up and talk about later. Okay, thank you very much. I think that's it. And I would like to welcome the second speaker, Agata Konieczna, with her title, The Binding Mechanism of Chaperone Proteins, HSP70 and JDP class B to the surface of protein aggregates.
Okay, so hello everyone. Today I would like to present about my master project titled The Binding Mechanism of Chaperone Proteins, HSP70 and JDP class B to the surface of protein aggregates. So proteins, to perform their function, needs to obtain their native conformation. However, under conditions of stress, like for example heat shock or mutations, they can lose their native structure and misfold. And those misfolded states can form dangerous amorphous aggregates or highly structured amyloid fibrils that are connected with neurodegenerative diseases, like for example Alzheimer or Parkinson disease. And to prevent those dangerous structures, organisms developed chaperone proteins that are able to disaggregate those amyloid fibrils and amorphous aggregates and also refold misfolded proteins to their native conformation. And one of the most known chaperone protein is HSP70 that cooperate with HSP40, and this is its canonical cycle. Today I will be focusing more on its yeast homologue SSA1. So firstly, cis1 recognize misfolded protein and transfer it to the SSA1 protein. And this interaction initiate ATP hydrolysis. So SSA1 bound to ADP close its alpha helical lead and trap the substrate inside of the substrate binding domain. Then after switch from ADP to ATP, alpha helical leads open and release a native protein. So as we can see, interaction between those cis1, SSA1 and aggregates is crucial for this cycle. However, exact molecular interactions remain not that clear. So in order to form an active complex of cis1 and SSA1, they need to bind in two binding sites. At least right now it is believed that they need to bind between beta SD1 domain and EVD motif of SSA1 and between J domain and nucleotide binding domain of SSA1. And as I said at the previous slide, canonically cis1 recognize and transfer the substrate, and then SSA1 interact with aggregate by substrate binding domain. So in my master project, I wanted to answer a question: what are the requirements for cis1 SSA1 complex to bind with the aggregate? Firstly, I wanted to see if cis1 alone can bind to the aggregate, and then how this interaction would change after forming a complex with SSA1. And to answer that question, I constructed SSA1 variant that cannot bind to the substrate. So then I can see only effect of cis1 within this complex. So I substitute valine for phenylalanine in position 435. And after I constructed a vector, I overexpress it in transformed Escherichia coli cells. Then I purify it on nickel column. And to get rid of SUMO and histag, I digest it with ULP1 protease. And at the end, at the ATP agarose column, I was able to confirm that my protein is active, because SSA1 has ATPase activity, so it's bound to the ATP molecules that are connected with ATP agarose column. And at the end, I receive purified variant with concentration about 8 micromoles. So in order to monitor interaction between this complex and aggregate, I used biolayer interferometry. And this is an optical method that allows us to monitor interaction between myomolecules in real time. So to do this, I firstly denaturated luciferase protein and immobilized it to the BLI sensor. Then I incubated the aggregate with mixture of chaperone proteins. And at the end, biolayer thickness indicates how well or if chaperones bind to the aggregated substrate. And here is a positive control, as we can see, association and dissociation. This is a wild type complex of cis1 and SSA1 wild type. And here we can see cis1 alone, and we see that there is no binding to the aggregate. And this is very surprising considering its canonical role of recognizing and transferring the substrate. Then when I added SSA1 variant that cannot bind to the substrates, there is still no binding. So this suggests that binding of SSA1 is crucial for this complex during the binding to the aggregate. So next part of my master thesis focused more on interactions between cis1 and SSA1 in order to bound to the aggregate. So as I said, right now it is believed that cis1 needs to be bound with SSA1 in 2 binding sites, here and here. And I wanted to investigate what would happen if I delete EVD motif. So as you can see, cis1 with SSA1 DEVD do not bind to the sensor at all. However, there was an interesting result. When we mixed cis1 with SSA1 wild type and SSA1 DEVD, the binding is the fastest and the strongest, even stronger than cis1 and SSA1 wild type. And we can see that cis1 with SSA1 deletion of EVD, there is no binding. So that suggests that maybe this complex bound somehow different, and right now this is an object of experiments in our laboratory. So overall, my experiments shows that cis1 alone do not bind to the aggregate, and also in complex with cis1 and SSA1, it doesn't change, and its interaction between SSA1 and aggregates is like the most strongest force that holds them together, and also that deletion of EVD motifs enables this construct to bind with the aggregate. So thank you very much. I would like to thank my supervisor and all my lab members.
Okay, thank you for the presentation. Are there any questions from the audience?
So I have a question. How exactly did you obtain this SSA1 variant?
Okay, thank you for your question. So first of all, I designed primers that allows me to perform PCR and site-directed mutagenesis, where I did this substitution from valine to phenylalanine. Then, after PCR, I digest my PCR products with DPN1 enzyme, so I received only mutated products. Then I transform it to the Escherichia coli cells, isolate the plasmid DNA, and send this for clonal analysis. Yes.
Okay. Any more questions?
So the recognition of substrate depends on a single amino acid residue, this valine residue. So how come that, is it known how they bind the substrate?
I know that previously the experiments was done when they wanted to see which parts of SSA1 bounce with the aggregate, and one of the most important amino acids is valine, yes.
Okay, but is it direct or indirect effect? So is it involved in the binding, or is it affecting the structure of this domain, for example?
I think it's involved, but I'm not sure.
Okay. I think that's it. Thank you very much. If there are more questions, great. And then I would like to welcome the third speaker, which is going to be Victoria Viernikuyskaya. I hope you say this correct. She's going to present to you a comparison of prokaryotic and eukaryotic nucleotide exchange factors in refolding of proteins from aggregates.
Today I want to present a presentation about my project titled A Comparison of Prokaryotic and Eukaryotic Nucleotide Exchange Factors in Refolding of Proteins from Aggregates. To function properly, a protein must be in its native state. This is challenging due to the presence of stressors like high temperature that lead to protein misfolding and aggregation, which is harmful to a cell. To avoid this, the cell has developed various protein quality control systems, which one of them is molecular chaperones. If aggregate forms, the cell has two ways to eliminate it: by degradation or disaggregation, which can be done by HSP70, HSP100 chaperone system. In my project, I studied the process of protein recovery from amorphous aggregates, which involves several steps, including aggregate remodeling, disaggregation, which when single polypeptides are released from the aggregate, and the folding into native conformation. This process can be done by HSP70 protein with its two cochaperones, J-domain protein and nucleotide exchange factor, and HSP100 protein. In my project, I studied bacterial system and yeast system. HSP70 is made up of nucleotide binding domain and the substrate binding domain with the lid, which closes on bound substrate. In ATP bound state, lid is open, so substrate can easily bind to and unbind from HSP70. In ADP bound state, lid is closed, trapping the bound substrate. HSP70 functions in ATP dependent cycle, and the cycle starts with HSP70 in ATP bound state. Then J-domain protein and substrate interaction with HSP70 trigger ATP hydrolysis, generating ATP bound state. Then NEF promotes the exchange of ADP to ATP, resulting in substrate release, and HSP70 is ready for a new cycle. Eukaryotes have only one family of nucleotide exchange factors, GRPE. Eukaryotes have three families. Proteins of these families have different structures, but they share a function. They accelerate ADP to ATP exchange in HSP70 by stabilizing an NBD conformation with an open nucleotide binding cleft. In my project, I studied GRPE protein and a member of HSP110 family, SSL1 protein from yeast. The aim of my study was the determination of the role of bacterial and yeast nucleotide exchange factors in the recovery of proteins from aggregates. I started with luciferase refolding assay. In this assay, a native luciferase was denatured, and then aggregates were prepared by dilution and incubation of unfolded proteins. The refolding reaction was started by adding aggregates to the mixture of chaperones. And the refolding efficiency was measured by the light emitted in the reaction with luciferin. Using this assay, I tested how NEF affects the process of luciferase refolding. In bacterial system, increasing NEF concentration improves the refolding reaction. In yeast system, however, increasing NEF concentration decreases the efficiency of refolding. So I set out to investigate at what stage nucleotide exchange factor influence the luciferase refolding process. I used a biolayer interferometry technique, which also allows the measurement of molecules binding efficiency, but I a little bit modified it. And also I used fluorescent labeled luciferase in this assay. So first I prepared aggregates of this luciferase on Ninta surface and incubated them with a mixture of chaperones. During association step, chaperones interact with aggregates, release polypeptides from them to the buffer so they can fold into their native structure. Then I measured the amount of luciferase released from the aggregates by measuring fluorescence and folding efficiency using the reaction with luciferin. In bacterial system, both fluorescence intensity and luciferase activity are higher when NEF is added to the reaction. This indicates that GRPE enhances the process of polypeptide release from aggregates and their folding. However, in the yeast system, fluorescence intensity is different for different NEF concentrations, but luciferase activity is almost the same. For 0.1 micromolar concentration of NEF, there is an improvement in the process of polypeptide release from aggregates, but at a higher concentration of NEF, there is a decrease. So my results suggest that GRPE is essential both at the early stages and the final stage of protein recovery from amorphous aggregates. And the HSP100 intense effect is concentration dependent, and it is primarily significant at the early stage. So thank you for your attention.
Thank you very much. Do we have do we have questions from the audience for her? So over there and then.
So my question is, because you see different effects of F in bacterial and yeast system, and if you in yeast system, for example, if you have equivalent amounts of HSP70 and NEF, you see that here. So the amount of recovered luciferase is lowered. Can you explain maybe why do you think such effect you observe?
You mean 0.1 concentration or 1?
No, equimolar. I mean 1 micromolar NEF, 1 micromolar HSP70.
In my essays, I used 2 micromolar concentration of HSP70.
Okay.
Yes. But here, for example, I can see this decrease.
But still you see the inhibitory effect, yeah? So do you have a hypothesis what's going on? I mean, what is nucleotide exchange factor doing differently in comparison to bacterial system?
I think that in that GRPE protein, so NEF of bacterial system, functions only like as a nucleotide exchange factor, so it promotes this exchange of nucleotides. But in yeast system, SSR1 protein its structure is similar to HSP70. It also has nucleotide binding domain and substrate binding domain. So I think that at higher concentrations, it can just compete in binding to the substrate with HSP70. So it binds to substrates, and the HSP70 has low places to bind to aggregate.
Okay, the second question was here. It's still valid?
Yes. Okay. I wanted to ask why there are differences in polypeptide release from aggregate efficiency, like in SSA1 in like different concentrations.
So it is a similar question, and yes, it is all about the competition between, I think, between these proteins.
Okay. Thank you very much. I think that's it. Thank you for the presentation. And then the third person from the Laboratory of Protein Biochemistry is the speaker Mikko Wairei, who's going to tell you about the purification and biochemical characterization of human RAB6B protein variants suspected to be pathogenic.
Thank you very much. Hello, everyone. The topic of my master thesis is purification and biochemical characterization of human RAB6B protein clinical variants. It was done in a laboratory of protein biochemistry under the supervision of Dr. Szymon Zienkiewicz. RAB6B is a member of small GTP binding protein family, and in its active conformation, it functions as a molecular switch, activating the retrograde vesicle transport in the brain, which is necessary for proper neuron formation. it cycles between the inactive and active forms defined by the presence of the nucleotide in the nucleotide binding site. When GDP is present, the protein is in its inactive conformation, and when GTP is present, the protein is in its active conformation. Because this process is highly controlled in the cell, there exist GEFs and GAPs. GEFs are guanine nucleotide exchange factors, which are proteins that help activate the RAB6B protein, and GAPs are GTPase activating proteins which deactivate the protein in the cell. As I've said before, this protein is very important for the neuron formation in the brain. That's why any mutations may have disastrous consequences for the cell. Important for my master thesis, there are 3 variants: the wild-type variant and 2 clinical variants. The first clinical variant, RAB6BA171E, is a clinical novel variant that was discovered in Warsaw in a microcephalic patient that suffers from developmental issues and microcephaly, and we think that the mutation may be the reason for it. As you can see here, this is the structure of the RAB6B protein, and highlighted in red, we have the 171 amino acid. This is the place where the mutations occur that I'm investigating. And as you can see, it is away from the GTP binding site. That's why we weren't focusing on the activity of the protein, but rather on the stability of it and the secondary structures. Also important are the magnesium ions that are necessary for the GTP binding, and this fact is necessary for one of the assays later. The aim of the study is the optimization of the overexpression process of RAB6B variants, as well as preliminary biochemical characterization of 2 clinical variants, focusing on RAB6BA171E, as I said before. So first thing that we did was we purified the protein. Here we can see the overexpression of the protein pre and post. After confirming that the overexpression took place, we were able to purify the protein. At first using the HISTRAP XL column, which uses the affinity chromatography, and the HISTAC present in the protein. After preliminary purification, we digested the protein with TEF to remove the HISTAC. To further remove the HISTAC from the sample, we used once again HISTRAP XL column, and to finish the purification process, we used size exclusion chromatography. We repeated this process for all 3 variants, and we got 3 protein variants that are very clean, as you can see, and we calculated the concentration using densitometry. After confirming that the protein samples are clean, we were able to do the measurements, the secondary structure measurements, at first using the circular dichroism. Here you can see that every protein is folded, and we can see small differences between the secondary structure of each variant of the protein, with the biggest outlier being RAB6BA171E. Another measurements using the circular dichroism method were the melting curve measurement of the variants. Here in the temperature gradient, we were able to calculate the point in which half of the protein is folded and half of the protein is misfolded. And we were able to calculate this temperature to see the stability of each of the variants. As you can see, the most stable variant of the protein is RAB6BA171V clinical variant, and the least stable is RAB6BA171E clinical variant. Also, important thing to note here is that those temperatures are above the temperature of the human body, around 37 degrees, but still we can see the differences in the stability between the variants. Another experiment that we did, which was quite similar, it used the same buffer, was the tryptophan fluorescence measurements, melting curves. Tryptophan is a fluorescent amino acid. We can calculate the fluorescent intensity of tryptophan in the temperature gradient. As the protein unfolds, the fluorescence intensity changes, and we are able to plot that on the graph. From the graph, we are able to calculate the inflection temperatures, once again comparing the stability of the variants. The results are similar to the previous results from circular dichroism. Once again, RAP6B-A171V is the most stable variant, whereas the least stable variant is once again RAP6B-A171E variant. Another measurements using tryptophan fluorescence assay were the melting curves with EDTA. EDTA is used to halate magnesium ions from the protein, which, as I said, are necessary for the GTP binding. So here we have the baseline inactive form of every protein. And here the stability changes. The most stable variant right now is, in this experiment, is the wild type variant, and the stability of RAP6B-A171V dropped significantly. Those two variants have similar stability in this assay, and once again, the least stable variant is A171E clinical variant. So, to sum up, protein samples of all three variants were overexpressed and successfully purified. Secondary structure analysis showed that all variants are folded and that there are small structural differences between them. Stability measurements of the variants reveal high unfolding temperatures with differences between them, and removal of magnesium ions show bigger differences between the inactive variants, with RAP6B wild type being the most stable one. Thank you for your attention.
Thank you very much. Any questions from the audience regarding that presentation? I think there's one. Yeah, I will bring it. You can decide if you use it or not.
Thank you. Thank you for a nice presentation. What would be the next step now?
The next step would be to finalize the preliminary characterization, of course. And to do that, we would need to, in my opinion, do the activity assays for the proteins. But for that, we would need the GAPs and GEFs to help with the GTP hydrolysis. And then we would be able to have more concrete evidence for the variants. So yeah, this would be the next step.
Thank you.
Okay, any more questions from the audience? There's one.
Do you think of testing the stability with nucleotides, GTP or GDP?
So did we think about the stability with GDP or GTP?
Have you tested with or without, and what?
Okay, so in this assay we test without GTP, so basically like with GDP, but we had assays that, for the sake of time, I wasn't able to show here, but we also tested with excess of GTP, because if we incubate this protein with GTP and add magnesium ions as well, we are able to, over time, get all the variants to have GTP in the structure. So yeah, we did those measurements, but unfortunately I wasn't able to show them, and they are quite similar, yield similar results to this one. So yeah.
Okay. Any more questions from the audience? No, I don't think so. Thank you very much once more.
Thank you.
And as a last speaker, this time again from the Laboratory of Photobiology and Molecular Diagnostics, I would like to welcome Patrycja Pikulik, and she's going to tell us about the assessment of the risk of co-selection against antimicrobial blue light and thermal stress in Escherichia coli.
So hello everyone, my name is Patrycja Pikulik, and today I would like to talk about my master thesis, which is titled Assessment of the Risk of Co-Selection Against Antimicrobial Blue Light and Thermal Stress in Escherichia coli. I'm writing the thesis under the supervision of Dr. Aleksandra Rapacka-Zdończyk in the Department of Photobiology and Molecular Diagnostics. So let's start from introduction. Escherichia coli is one of the major foodborne pathogens, and in food industry, thermal treatment is widely used technique for food preservation as well as for decontamination of production lines and food packaging. But this technique has some disadvantages, like nutrition loss, changes in flavor and texture of treated food, and also it requires a lot of energy. That's why currently there is high interest in adding to the chain of food production some non-thermal methods like antimicrobial blue light. But what we have to remember about is that external stress leads to a bacterial defense response and development of stress tolerance, which can make bacteria also more resilient to other stressors, and such a phenomenon is called cross-adaptation. And from the previous studies conducted in our lab, we already know that E. coli can develop tolerance to ABL, and we also know that mutants lacking genes encoding DNA-J and DNA-K heat shock proteins are hypersensitive to ABL. That's why it's my master thesis. I wanted to try answering the question, is there any risk of E. coli cross-adaptation to temperature and ABL? So the first aim of my study was to obtain E. coli population tolerant to heat stress and another population tolerant to ABL. To further answer the questions, is the heat-tolerant population also tolerant to ABL, and opposite, is the ABL tolerant population also tolerant to high temperature? I had also two extra aims, which were assessing effects of different culture temperatures and thermal pre-incubation on E. coli K12 sensitivity to ABL. So moving to the first aim, to obtain tolerant population, 10 cycles of sublethal treatment of E. coli K12 with temperature or ABL and regrowth in the fresh medium was carried out. And what I mean here by sublethal treatment, it is a dose causing 90 to 99% decrease in bacterial survival. It is 1 to 2 decimal logarithm drop in survival rate, and it was followed by 5 cycles without any treatment to confirm that this tolerance is stable. So it is due to mutations, not due to any temporary gene expression changes. And population obtained in such way were then exposed to the stressors, and the average survival rate was calculated using a serial dilution method. And the results of it we can see here on the graphs, but also in numbers. So I can say that this part of my experiment was successful in both cases, temperature and ABL. We can see that the populations after 10 sublethal treatments have much higher average survival rate compared to the control, which is the green line here. And also the tolerance is quite stable. Only in case of temperature we can see a small drop, but still the tolerance here is unquestionable. So let's move to co-selection. Reminding the first question was, is the heat-tolerant population also tolerant to ABL? And basically I can say no, it's not. There are actually 3 doses where statistically significant changes were obtained. Those are 2 increases and 1 decrease in survival rate, but overall it can be explained as some uncontrolled experimental variables. And the opposite question was, is the ABL tolerant population also tolerant to high temperature? And here I can say yes. The ABL tolerant populations had higher average survival rate compared to the control, which is the green line here, for all the 4 tested temperatures. The differences you can see also here in numbers, but what should be noted here is that those results did not come close to the results of temperature tolerant populations, which had increase in survival rate around 3 decimal logarithms. But some interesting results were also obtained in the additional tasks. And to clarify it, here I was not no longer working on those tolerant populations. They were just normal E. coli K12 populations. So here we can see a graph showing response to ABL of 2 populations, one cultivated overnight in 37 Celsius degrees and the other one in 42 Celsius degrees. And we can clearly see that the average survival rate of the population from higher temperature is like higher than the standard temperature control. And such differences are even more visible in the last task, where bacteria were pre-incubated in 50 and 52 Celsius degrees for 10 and 20 minutes, and the populations which were pre-incubated are less sensitive to ABL compared to the control, which is the black line here, and it is especially visible for 10 minutes in 50 Celsius degrees pre-incubation, where the increase reached 2.55 decimal logarithm, which is pretty close to ABL tolerant population. So to sum it up, E. coli K12 develops tolerance to ABL and to thermal treatment as well. The heat-tolerant population shows no altered sensitivity to ABL, but ABL-tolerant population shows reduced sensitivity to heat stress, and also overnight cultivation at elevated temperature, as well as thermal pre-incubation, makes E. coli less sensitive to ABL. Thank you so much for your attention.
Thank you very much. And any questions from the audience for our last presenter? Someone has a question again? Okay.
Thanks for the presentation. I was curious because you showed that deletion of DNA K causes cells to be sensitive. Actually, deletion of DNA K, from what I think, is lethal. So do you know how did they show it? And the second question is, because you saw differences between the stored population of the cells that are resistant, right, and the cells that were pre-incubated. Do you have any explanation for that? Can you think of any explanation of why did you see the differences between this pre-treatment and this heat-resistant cells in regard to sensitivity to blue light?
Okay, starting from this first question, it's actually not my publication, so I was not taking part in those experiments. But as I remember, those were just knockout mutants. So somehow I think they got the results. I cannot explain it in details. This is just what I remember from the publication. And the second question, to clarify, sorry, what differences?
How do you think of explaining the differences between thermal tolerant cells sensitivity to blue light, or rather lack of this, and the pre-treatment sensitivity?
Okay, so in this first part of my experiments, I was rather showing the mutations in the genome of E. coli. And in the second part, where there were only pre-treatments, it was a change of expression. So basically what we see from here is that most probably this resistant or tolerance actually to ABL is due to changes of expressions of some genes encoding heat shock proteins, most probably.
By curiosity, did you sequence the strain before and after experiment?
No.
Good. Okay. I think that's enough questions. Thank you very much. And that concludes the third session. And now there's a small coffee break, 20 minutes. I think it's again in the room over here. And the next session starts at 15:40, 22:04.
Okay, welcome back to the last session of the day, and we have exactly 8 speakers lined up. The first speaker is Ms. Yulia Bilinska, who will be talking about the application of mass spectrometry for quantitative determination of cortisol in diverse biological matrices. So, Yulia.
Good morning, everyone. My name is Julia, and today I would like to present you my thesis of my master thesis, application of mass spectrometry for quantitative determination of cortisol in diverse biological matrices. My supervisor is Professor Stanisław Ołdziej, lab supervisor, PhD student Michał Puchalski, and I'm conducting my research at the Laboratory of Biopolymer Structure. Let's start by give you a brief background of my work. The cortisol is a glucocorticoid hormone mainly produced by the adrenal cortex. As part of the body fight or flight responses, cortisol is released during stressful times to give our body natural energy boost. Cortisol secretion is controlled by the hypothalamic pituitary adrenal axis, and the fundamental role of cortisol is to maintain homeostasis during physiological metabolic changes and to adopt the organism to a survival state following to the stressor. This is achieved through the regulation of metabolism, increasing gluconeogenesis to ensure an adequate energy supply, influencing the circulatory system to regulate blood pressure and exhibit anti-inflammatory effects. It is also worth mentioning that I started my work by trying to extract cortisol from the archaeological teeth and dental calculus samples. Teeth are some of the best preserved remains of organisms from a bioarchaeological perspective, and the cortisol itself is a valuable source of information about health and adaptive strategies on earlier human communities in the face of changing living conditions. Unfortunately, it was not possible to obtain reliable results. The scientific objective of my study is to assess the effectiveness and reliability of mass spectrometry methodologies, quantifying free cortisol concentration, considering the inherent complexities and heterogeneity of different biological matrices. In my study, I used 4 matrices: blood plasma, saliva, follicular fluid, and urine, and focused on free cortisol, a biologically active molecule that is not bound to any protein such as albumin or globulin. As you can see on the slide, it is only 5% of total cortisol content, so relatively low concentrations are expected. Classical methods for free cortisol testing include extraction using zinc sulfate. This procedure is quite time-consuming. New ions are introduced and requires additional desalting steps. To simplify the procedure, I use centrifuge filters to remove proteins from such complex biological matrices. These filters have a cut-off point 10 kilodaltons, so most proteins will remain on the filter, while the compounds below 10 kilodaltons will be in the filtrate. All samples were analyzed using mass spectrometry in multiple direction monitoring mode. There is only important to note that cortisol level fluctuates in diurnal cycle. Therefore, the concentration in individual matrices may differ due to the unknown time of sample collection. The first chart shows the free cortisol concentration in blood plasma. The concentration range between 1 and 12 nanograms per milliliter, and according to the literature, they fall within the clinical references ranges for healthy adults. The second graph presents the free cortisol concentration in saliva, and the concentration between 1 and 9 nanograms per milliliter, and they also fall within the clinical references ranges. The third chart shows the
free cortisol concentration in follicular fluid. Despite the lack of established references ranges for this matrix, the results are consistent with expectations, showing the comparable concentration to those in the plasma and saliva. The final chart presents the free cortisol concentration in urine. The concentrations are significantly higher than in the other matrices, and exceeding even 120 nanograms per milliliter, and which aligns with the literature, indicating that cortisol in urine is primarily in the free form, as bound cortisol is not filtrated by the kidneys. Due to the low concentration of free cortisol, derivatization was also investigated. This was done to lower the quantification threshold and improve analytical characteristics. Each sample was done in 2 repetitions with and without derivatization using mass spectrometry. Derivatization using Girard-Tira agent. The Girard-Tira agent previously used for derivatization of other steroid hormones, and all the samples were then analyzed using mass spectrometry in multiple reaction monitoring mode, just as before. On this slide, you can see the concentrations obtained in saliva. The brown bars represent the cortisol without derivatization, and the black bars represent cortisol after derivatization using Girard-Tira agent. As you can see, the concentration after derivatization is significantly higher than those in the cortisol without derivatization, even 4 times higher. And these results are unreliable. Expected results were similar to or lower than the cortisol without derivatization due to the incomplete reaction of analyte. In summary, the conducted research has demonstrated that extraction methods using centrifugal filters provide comparable results for determining free cortisol to the classical method and can be applied in various biological matrices. However, Girard-Tira agents proved to be ineffective for cortisol measurement. Further studies using other derivatization reagents could extend the research potential of developed method and facilitate analysis of samples with very low concentration, cortisol concentration, such as archaeological samples. And thank you for your attention.
Thank you, Julia. Do we have any questions for Julia? Yeah.
Thank you for the presentation. I might have missed it, but did you use any controls, maybe a sample with known cortisol concentrations to benchmark your method?
First of all, I prepared the standard curve of cortisol, and due to them, I calculate the concentration in appropriate samples. So,
I have a follow-up question, because I recently read that there might be some sensor cells for sensitive to cortisol with regard that expression of the reporter was dependent on the presence of the cortisol. Have you read about it? Maybe to compare the sensitivity between your method and such available methods of some sensors or biosensors?
I think that I have no knowledge about this sensor, so I don't know.
Any other questions?
A short one. So why would you like to measure cortisol in archaeological samples? So what knowledge can it give to us?
as I said at this slide, cortisol can give us important information about health and adaptive strategies of aerial human communities. So we can know more about exactly adaptive strategies.
Asian people were more stressed than us or not? Or
for example.
So how persistent is cortisol in the tissues? I mean, if you have teeth, and is there any, does cortisol is preserved within the tooth?
It's a lot of publication where scientists research cortisol in hair and teeth, and it is possible, and it can be preserved at the tips.
Yeah, I think that's all we have time for. So thank you, Julia, once again. The next speaker is Magdalena Labuda, who will be talking about synthetic NTPases and nucleases based on TRP-zip motif. Magdalena.
Hello everyone, my name is Magda, and today I would like to present you the presentation about my master thesis. The title of my master thesis is Synthetic NTPases and Nucleases based on tryptophan zipper motif, under the supervision of Professor Stanisław Ołdziej and his PhD student Marcel Thiel in Biopolymer Structure Laboratory. Let's start by explaining the purpose of my work. The purpose of my work is testing short peptides based on tryptophan zipper motif capable of catalyzing the hydrolysis reaction of nucleic acids, mainly DNA, and then comparing them depending on the environment. Environment like pH, different concentration, time of incubation, and temperature. Why did we decide to conduct such a research? There were indications in the literature that peptides were capable of hydrolyzing nucleic acids. Also in my lab, they tested hydrolysis of ATP via peptides. Thanks to the stable structure provided by tryptophan zipper motif, only 12 amino acids peptides are able to perform work of enzymes, which are huge, large protein complexes. What's more, the costs of proteins are high, and synthesis is complicated, so we are looking for a replacement. For my research, I use capillary electrophoresis. It's an analytical technique that separates ions based on their electrophoretic mobility with the use of an applied voltage. This method is one of the fastest developing techniques from which we quickly obtain high resolution results. Capillary electrophoresis could be used to separate a wide range of different molecules, such as organic acids, drugs, biopolymers, and a lot of more. Here we see a diagram of capillary electrophoresis system. Everything takes place in a glass capillary of a specific width and length from anode to cathode. In my work, I checked 5 peptides. The most important is TRPZIP2, because this is the basic literature peptide based on tryptophan zipper motif, and 4 of its variation. For a better visualization, I created this illustration of the structure of this peptide. And the first one, the main peptide, TRPZIP2, that contains 4 tryptophan residues that create a so-called basket structure that allows to bind the DNA molecule. In the next peptide, TRPZIP6, 2 tryptophans are changed to different amino acids that may affect the ability to bind to the DNA molecule. We know that lysine plays the main role in a hydrolysis reaction. So in the next 2 peptides, there were 1 and 2 less methyl group in this area, which shorten these loops. These 4 peptides undergo amidation and acylation at the end, so the last version, the last peptide, has a normal carboxyl and amino groups at their ends. So let's move on to to my selected for you results. We look at how DNA hydrolysis occurs over time after incubation at 40 degrees for 4 days with peptides. The first spectrum shows the DNA separation. It's our control sample without peptides at time 0 in neutral environment. Here we have also the sequence of our DNA. It's a short fragment. We don't use the purest DNA, so we see some contaminants at the beginning, but we focus on the main peak. This is the shape of our DNA that we have to remember. The next slides show the same sample. It's our control sample after 4 days of incubation at 40 degrees. We see a similar peak as in the previous slides. Nothing is happening here. So thanks to this, we know that our DNA is stable and nothing happens to it spontaneously. The next slide shows the DNA separation with addition of TRPZIP2 ORN peptide at time 0 in neutral environment in concentration ratio 1 to 10. We see a similar peak as in the case of pure DNA. However, after 4 days of incubation at 40 degrees, we see a significant differences. Our main peak has decreased and we see shorter fragment of cut DNA at the beginning. That means that our DNA hydrolysis has occurred. The same situation we have with addition of TRPZ6 peptide. At time 0 in neutral environment, we see the same peak as in the case of pure DNA. However, after 4 days of incubation, we see that our DNA hydrolysis has occurred. After conducting a series of experiments, we are sure that the right amount of peptide and properly selected conditions ensure the hydrolysis of single-stranded deoxyribonucleic acid, which was presented in the previous slides. Further research at checking the best hydrolysis conditions for individual nucleic acid may be the beginning of replacing enzymes with a small, stable peptides. In the future, we can test a larger number of different peptide variants and selected environmental conditions and choose the best one for each type of nucleic acids, DNA, RNA, single-stranded, double-stranded, and even, for example, depending on what type of experiment we want to use it for. And this is all. Thank you for your attention.
Questions? Any questions?
So those small nucleases, they're synthetic. So is it expected that they will be, for example, degraded in the biological material, or they should be stable in the biological material? Are there any proteases like expected to degrade it?
This tryptophan zipper motif is characterized by presence of tryptophan residues. And this peptide are, thanks to this peptide are stable and have a hairpin structure, a three-dimensional structure. And thanks to this, we know that these small peptides are stable in a high temperature, even 70, 80 Celsius degrees. So it's a very stable structure.
Do they occur in, let's say, biological materials? So are they like synthetically constructed, or are they like following something that exists in nature?
I don't know that these peptides are in nature, but we synthesize it in the lab. So,
thank you.
Oh, one more question there.
Yes, I can speak out loudly.
Don't have time. So can we take this offline because we're running out of time? We're running out of time, sorry.
We don't have time.
Yeah. Okay, thank you, Magdalena.
Thank you.
So the next speaker is Mr. Viktor Jarecki. He will be talking about the impact of CD1A distribution in tolerogenic dendritic cells on antigen-specific T-cell responses.
Hello everyone, my name is Viktor Jarecki, and today I would like to show you my master thesis titled The Distribution of CD1A molecules on antigen-presenting cells upon exposure to permissive and non-permissive ligands. The study is conducted in Laboratory of Experimental and Translational Immunology under the supervision of Professor Danuta Gutowska-Osiak. So first, when there is immune response, we have interaction between antigen-presenting cells and T cells. And during this interaction, we have education of T cells about the antigens. This happens through the molecules that presents antigens. While peptide antigen presentation occurs with MHC class 1 or class 2, the lipid antigen presentation involves CD1 molecules, which are cell surface glycoproteins known for presentation of lipid antigens to T cells. And in this, human cells express 5 isoforms of CD1 molecules, CD1A, B, C, D, and E. But in the study, we are focusing only on CD1A molecule, mostly because it's relevant to the skin. So lipid antigens can be divided into 2 groups: permissive, which induce T-cell activation, and non-permissive, which inhibit T-cell activation. And the aim of the study is to investigate the cellular distribution of CD1A molecule in antigen-presenting cells upon exposure to various sources of lipids. So we have a model of antigen-presenting cells. K562 empty vector, which is our control, and K562 CD1A, which have been transfected with CD1A molecules. And thanks to this, we can nicely observe distribution levels on their surface. CD1A is also the only antigen-presenting molecule that they have, so wherever we're observing is relating to them. And we also confirm that we don't have any CD1A signal on the empty vector transfectants. So for the simplicity of this presentation, I'm just going to show the results for K562 CD1A. So the base of the study were those 2 cell lines, which I stained with either anti-CD1A APC antibody alone or additionally with anti-CD1A FITC antibody. Then the cells were incubated with various sources of lipids and taken for flow cytometry and imaging. And our sources of lipids were phospholipase A2 enzyme, which I will explain in a second, permissive and non-permissive lipids, and lipids contained within membranes of small extracellular vesicles. So phospholipase A2 enzyme is an enzyme which pulls out membrane-associated lipids, cleaves them, and creates antigens that are going into CD1A groove, and later on can be presented. And first, we wanted to see the distribution of CD1A upon exposure to PLA2 as the most commonly known source of lipid antigens. I wanted to see time-related trajectory of CD1A. And we can see a decrease starting from 2 hours, which indicative of CD1A internalization, which reduces staining on the surface. But we don't, from the flow cytometry setup, we don't know exactly the distribution of the molecules, where they are, so we did imaging. And from this imaging, we can see the differences in the distribution, even if you don't see decrease from the signal yet. So after 50 minutes of incubation with PLA2, we can see that CD1A is localized only on the surface of the cell. And after 1 hour, the CD1A is starting to internalize. After 2 hours, the signal is coming only from the inside of the cells, and after 24 hours of incubation, we don't see any signal. So APC CD1A signal is showed as a gradient of gray value, gradient of gray value, the more blue, the less intense the signal is. And the peak showed in the central part confirms that we have internalization of the signal. But from those pictures, we could see that after 1 hour, the stained whites were pulled inside, but we didn't know what happens on the surface. So we did FITC staining. And we can see that after, if we add another antibody after 1 hour of incubation, the staining is showing on the surface. So that indicates that some of the CD1A are recirculated back, or the new ones are synthesized and brought up to the surface. Our second source of lipid antigens were 4 lipids, 1 non-permissive and 3 permissive. And from the results, we can see that lyso-PC, which is a permissive lipid, induces internalization really quickly because we're losing the signal very fast. With the time, with the other ones, we can see slight differences, but they are rather small in comparison with lyso-PC. And our third source of lipid antigens were small extracellular vesicles, which are phospholipid membrane organelles that take part in intracellular communication. And previously, it was shown that SEVs contain lipids in their membranes, and we did lipidomic panels on SEVs from different cell lines. And based on the results, we chose those 2 cell lines, K562 and HEC293T. And the cells were cultured for 48 hours in EV-depleted medium, and SEVs were isolated through serial ultracentrifugation protocol. And the results from lipidomic panel also showed that HEC293T had more non-permissive lipids, which indicates that they reduce expression levels slower, because those are the stabilizing lipids. And the results showed that lipids derived from SEVs from HEC293T cell line induce a lower degree of internalization in comparison with K562, which confirms the results from the lipidomics. And so the conclusions are that after antigen exposure, CD1A undergo trafficking inside and back to the surface. Various sources of lipid antigens can promote internalization of CD1A molecule. Lyso-PC is most potent to induce internalization, and lipids from SEV's membrane induce less pronounced changes, likely because they contain mix of permissive and non-permissive lipids. We also want to confirm these results on different types of dendritic cells generated from healthy donor samples. I already have preliminary data from 2 donors, which are showing us the same pattern with lyso-PC as before. And we also want to test how CD1A levels can impact the interaction between APC and T cells. And at the end, I would like to thank my supervisor and the whole DGO lab team for helping me with my experiments. Thank you.
Questions? Questions for Adam?
So the first question is about phosphatidylserine. So is it a permissive or non-permissive lipid? This is like out of curiosity how our cells work. So phosphatidylserine, because this is quite specific for EVs, right? Because EVs contain PS addition, generally. So that's the first one. Like, do you know if this is a permissive or not? And the second is about, are there antibodies available for CD1A that can recognize the molecule only in the complex of lipid, with the lipid, like that would maybe be helpful in the assay. So are there antibodies that recognize not free CD1A, but only with complex with the lipid?
For the first question, I think it's a permissive. And with the other one, with the antibodies, I'm not sure if they are. They are.
Okay. Well, thank you, Victor. So the next speaker will be Mr. Adam Jałtuszewski. He will be talking about the analysis of gene expression of MHP and SARS-CoV-2 coronaviruses in the mammalian expression system. Thank you.
Hello everyone, my name is Adam Jałtuszewski. Key, and my topic is analysis of spike protein gene expression in MHV and SARS-CoV-2 coronaviruses in mammalian cells. My supervisor is Professor Krystyna Binkowska-Szewczyk, and I did my work in Laboratory of Molecular Biology of Viruses. The two coronaviruses that I'm interested in are SARS-CoV-2 and MHV coronaviruses. SARS-CoV-2, it causes COVID, a pandemic in 2020, over 700 million cases so far. And MHV is mouse or murine hepatitis virus, which is a model organism for coronavirus research in laboratories all over the world. And on the right, there's a scheme of structural proteins of SARS-CoV-2 and MHV viruses, but my main interest is spike protein of both. Also, I'm interested in synthetia formation, and there are two crucial factors for synthetia formation. First of all, there has to be a cell that expresses viral fusion protein. In my case, it's spike protein. And also, there has to be a neighboring cell with hostile receptor, for example, ACE2 receptor. And another crucial factor, here on the right, there's a scheme of spike protein. It consists of S1 subunit and S2 subunit that together create ectodomain of spike protein. And there has to be a cleavage between the subunits, S1, S2, and at the end of S2 subunit for activation of fusion peptide and synthetia formation. My aim of the study is checking whether the S protein itself alone has an effect on the cells, on their morphology and synthetia formation. And the research is part of a larger project investigating the cell-to-cell spread of viruses. And my workflow is divided into two parts. The first part is assessing the effects of transfection with plasmid coding the S protein of SARS-CoV-2, 3 different variants, on the morphology, cytoskeleton, and synthetia formation of the human cell lines and 1 monkey cell line with 2 methods, Western blotting and immunofluorescence. And the second part is assessing the effects of transfection with plasmid coding S1 subunit or ectodomain of S protein of MHV virus on the same aspects of the cells, but on murine cell line, which is mouse fibroblast LR7. Here are the results of the optimization of transfection with 2 different methods. One was lipofectamine 3000, second one jet prime. And according to those results, I selected the most efficient method of transfection for different cell lines. Some of the cell lines were already optimized before in the lab, and I obtained those results with a flow cytometry FACS, which is fluorescence activated cell sorting. And now I would like to go to part 1 of results connected with SARS-CoV-2 and human monkey cell lines. So here are Western blotting analysis results of SARS-CoV-2 as protein, and different cell lines have different results. For example, in A549 cell line, which is lung cancer cell line, I observed not much of a protein, but it also might be due to low transfection efficiency. Another example, in HEC ACE2 cell line, I observed a lot of spike protein, also different forms. In Wuhan and Delta variant of the spike, I observed also cleavage into subunits, which I did not observe in Omicron variant. And another example, in VIRO-E6 cell line, I observed spike protein for Delta and Wuhan, but no for Omicron. And immunofluorescence analysis, for each cell line there will be negative control, so the cells that were not transfected, and different variants of spike protein. For A549 cell line, I observed some expression of spike protein, but I did not observe any synthetia. And also in red there is protein spike, there is beta-catenin, so basically outer cell membrane, and in blue there are cell nuclei. In HEC ACE2 cell line, I observed a lot of spike protein in Delta variant, and also I observed big synthetia, so giant multinucleated cells, which I did not observe in Wuhan and Omicron. In human fibroblasts, it's similar. I also observed synthetia in Delta variant and a lot of spike protein, no synthetia in Wuhan and Omicron. And in VIRO-E6, I observed synthetia in Delta and Wuhan variants of protein, and in Omicron there was no synthetia formation. Part 2 results of MHV virus. Here I have less results because for a long time I was trying to construct 2 expression plasmids, one coding S1, one coding S ectodomain of spike protein of MHV, but it took me a lot of time because of some problems with, for example, restriction enzymes. So right now I only have these results, which are Western blotting analysis of S1 subunit and S ectodomain of spike protein of MHV virus. So right now I only know that there is production of spike protein in mouse fibroblasts, but I'm still waiting for immunofluorescence results. Right now I can show you a photo from another work by Natalia Stachura, which is doing PhD. And here it shows how mouse fibroblasts look after infection with MHV virus. And conclusions, when it comes to part one, so the S protein of SARS-CoV-2, all transfected cell lines produce S protein, but with variable efficiency. In some cases, protein S alone is sufficient to form syncytia, especially when there is cleavage into subunits. In the S protein of Omicron SARS-CoV-2 variant, the cleavage into S1 and S2 subunit seems to be very low or absent, and this also results in less syncytia formation. But the word seems to be is crucial because it also might be due to low efficiency of antibodies for Omicron variant of protein. And the S protein of Delta variant of SARS-CoV-2 has the greatest impact on the morphology of cells and syncytia formation. When it comes to MHV, I don't have many because I'm still working on it, but I know that construction of plasmids was successful and the mouse fibroblasts produced the S protein. And future plans, evaluation of the amount of ACE2 receptor and TMPRSS2 protease in the studied cells. This might correlate with syncytia formation. Also, transfection of mouse cell lines with plasmid encoding S protein of SARS-CoV-2 and vice versa, so like cross-species transfection. Also checking another protein, which is E protein, whether there is some kind of other effect with E protein, and comparing the results while infecting the cells with MHV. And that's everything. Thank you for your attention.
Have questions?
Hi, thank you. Great presentation. I have a question because you have used a lot of different cell lines. When I get the human cell lines, why have you also used the monkey one and the murine ones?
Yeah, so basically when I was choosing the cell lines, I tried to pick different various cell lines. For example, the A549 cell line is a lung cancer cell line. HEC is human embryonic kidney. I use fibroblasts to compare them with mouse fibroblasts. And VRO-E6 is a commonly used cell line for SARS-CoV-2 experiments.
So how divergent are the S proteins for these from these variants, and where the difference lie that could determine the different behavior?
Yeah, so like I observed, there are differences when it comes to production of the protein, when it comes to different variants. This might be due to many different mutations in different variants, because, for example, Omicron has a lot of mutations in spike protein, so that's why they give different results.
Yeah. Okay, thank you, Adam. Okay, the next speaker is Ms. Evelina Chodowiec. She will be talking about role of FGFR4 in proliferation and growth of triple-negative breast cancer cells and their response to PARP inhibitors. So, Evelina.
So I would like to present my master project, which title is Role of FGFR4 in Proliferation and Growth of Triple-Negative breast cancer cells and their response to PARP inhibitors. My supervisor is Dr. Dominika Piasecka from Laboratory of Molecular Enzymology and Oncology. Based on World Human Organization, breast cancer is the leading cause of cancer death in women and the most commonly diagnosed cancer in 2020. According to expression of biomarkers, including estrogen receptor, progesterone receptor, and human epidermal growth factor receptor 2, breast cancer consists of luminal A, luminal B, HER2 overexpression, and the most aggressive one, triple-negative subtype.
triple-negative breast cancer lacks expression of all biomarkers and commonly occurs in younger women. TNBC is associated with increased malignancy and mortality due to its high heterogeneity. And that is why finding effective targets for treatment is quite challenging. One of few valid targeted therapies are poly-ADP-ribose polymerase inhibitors. So PARP inhibitors suppress activity of enzyme PARP and blocks DNA damage repair, leading to tumor cell death. And this function is associated with mutation in homologous recombination repair genes, such as BRCA1 and 2 genes, which occur in almost 20% of TNBC patients. But there are growing evidence of effectiveness of this therapy in patients without mutation in BRCA1 and 2 genes due to BRCAness phenotype. However, PARP inhibitors can be used only in specific group of patients, so there is urgent need of finding other molecular markers and targets for TNBC treatment. And the attractive target seems to be the fibroblast growth factor receptor 4. FGFR4 is a member of tyrosine kinase receptor family, among with FGFR1, 2, and 3, and this receptor shares the least homology and contains the unique amino acid in its tyrosine kinase domain, the cysteine residue. And this change in sequence has enabled to design a specific inhibitors of FGFR4, such as roglitinib. But what's important, FGFR4 has unique role in metastasis and resistance to anti-HER2 therapies in luminal and HER2-positive breast cancer. But there is significant lack of knowledge about role of this receptor in triple-negative breast cancer. So taking into account all this information, the aim of this study was to evaluate role of FGFR4 in proliferation, growth, and response to PARP inhibitors of TNBC cells. Our research model were cell lines of triple-negative breast cancer, CAL51, MDA-MB453, and HCC1143. And each cell line is characterized as specific subtype of TNBC with mutation in BRCA genes or lack of it. What's more, MDA-MB453 cell line has constitutive activation of FGFR4. So in this study, our first goal was to check if FGFR4 is significant for TNBC cells proliferation by performing MTT test. And here we observe that CAL51 and MDA-MB453 cells proliferation fell after inhibition of FGFR4 by roglitinib, but only with use of high concentration of inhibitor. However, HCC1143 cell line cells proliferation did not change after 72 hours. After that, we tried to evaluate the role of FGFR4 in growth and colony formation of TNBC cells by using clonogenic assay. And after 2 weeks treatment with roglitinib, we confirmed that there were significant less colonies formed by CAL51 and HCC1143 cell lines after treatment with inhibitor, but there are no significant changes in growth in colony formation of MDA-MB453 cell line. So the unclear results of checking the proliferation after inhibition of FGFR4 by roglitinib made us wonder if knockdown of FGFR4 will change proliferation of these cells. And here we performed the MTT test, which compared proliferation of variant wild type and knockdown of FGFR4. And these results confirmed that FGFR4 is not significant for TNB cells proliferation. The second part of our study was to evaluate FGFR role in response to PARP inhibitor treatment. First, we wanted to compare proliferation of variant wild type and knockdown of FGFR4 after treatment with PARP inhibitor talazoparib. And here we observed that there were no significant differences in proliferation of each cell line. So to further evaluate if after a longer incubation with PARP inhibitor, there will be any changes of growth and colony formation of TNBC cells, we performed clonogenic assay. And after 2 weeks treatment with talazoparib, we observed that there were significant more colonies formed by cells with knockdown of FGFR4 than cells with variant wild type. What's more, HCC1143 cell line was sensitive sensitive to PARP inhibitor treatment, possibly due to BRCA phenotype. And this all results suggested that FGFR4 sensitizes TNBC cells to PARP inhibitor treatment. Lastly, we wanted to check our previous results out in 3D cell culture, so we used soft agar colony formation assay. And here we observe that cells with knockdown of FGFR4 form significant bigger colonies than cells with variant wild type. And these results confirmed our previous suggestion that FGFR4 plays important role in better response to PARP inhibitor treatment by TNBC cells. So our overall conclusions are that FGFR4 signaling is important in growth and colony formation of TNBC cells. What's more, FGFR4 sensitizes TNBC cells to PARP inhibitor treatment, what is independent of BRCA1 mutation status. And due to that fact, FGFR4 has potential to become a predictive marker of response to that therapy by triple-negative breast cancer patients. And that's all from me. Thank you for your attention.
Questions, please? Any questions? No? Okay, so you mentioned about the triple-negative breast cancer cell line that you were using. So what were some of the most important cell surface markers that we need to look out for in case we want to test, for example, you know, the response to, say, the survival of the, you know, the patient, or the overall survival, for example. If you want to know how many months or how many years the patient survives, like, so what are the kind of markers that you would want to look out for? Like, is FGFR4 could that be an important biomarker for that?
FGFR overexpression leads to like less survival of patients.
It's less survival of patients.
Yes, it's less
okay.
survival of patients, and mostly they're resistant to therapies.
Okay.
However, here we observe a little different function of FGFR4, because in our results, FGFR4 do not mediate resistance, but like is a marker of better response to this resistance, to this therapy.
Right. Thank you so much, Evelina. The next speaker is Mr. Wojciech Sadowski. He will be talking about the influence of FGFR4 on the response of LAR TNBC cells to CDK4/6 inhibitors.
Hello everyone, I'm Wojciech, and today I want to share with you the main aspects of my master's thesis, which I carry on in laboratory of molecular enzymology and oncology under full watch eye of Dr. Dominika Piasecka. And the name is the role of fibroblast growth factor receptor 4 in the response of luminal androgen receptor triple-negative breast cancer cells to CDK4 and 6 inhibitors. Don't worry, I will start with explanation what is the subject of my research. Let's start with the classification of breast cancer. As you can see, the triple-negative breast cancer is the cells are characterized by lack of 3 key receptors, like estrogen receptor, progesterone receptor, and human epidermal growth factor receptor 2. So because of the lack of that receptors, the triple-negative cancer is difficult to treat with therapy, because many of therapies are based on the mechanism against these 3 of receptors. And that causes the poor prognosis for the patients. Also, it's important that the tumor is not the homogeneous entity environment, but it's also some kinds of collection of the cells with different gene expression profiles, and that's how we can see here. Even the triple-negative breast cancer are also split for many of subtypes. And the subtypes which are important for us, is the hormone signaling luminal gene upregulation and it's characterized by overexpression of androgen receptors. That means even if we aim that the triple negative breast cancer is lack of estrogen receptor and progesterone receptor, we can name it luminal because androgen receptor is also from the family of receptors for the steroid hormones. That's why we can use the same therapy for the patient with the luminal cancer, like with progesterone and estrogen receptor positive. And in fact, we could also use the same therapy for the luminal androgen receptor triple negative breast cancer cells. And how does this work? So I can see, as you see, the inhibitor for cyclin-dependent kinase 4 and 6 cause the inactivation, the transcription factors, which are going to stop the cell in the G phase of the cell cycle, and do not allow it to go to the S phase. Then it's decrease the speed of proliferation of the cells. However, it's not so simple, because as you can also see, and as I mentioned, the tumor is not the homogenic environment. There are some kind of microenvironment, which are lots of many other cells, different than the cancer cells, which are stabilized, protect, and many other things doing to the tumor for the proliferation and other aspects. And we are mostly focused on the fibroblast growth factor receptor, which become from cancer-associated fibroblasts. These cancer-associated fibroblasts secrete, by paracrine mirroring, fibroblast growth factor receptor, which activate the fibroblast growth factor receptor, and start the cascade of the signaling. At the end of the cascade, the active fibroblast growth factor receptor causes the differentiation, proliferation, survival, and angiogenesis. However, one of them, fibroblast growth factor receptor, is a bit different from the other. That's why we are focused on them. Also, we are significantly know the less of the from the rest as in the aspect of triple negative breast cancer about the fibroblast growth factor receptor. So after that, that theory, we go to some research problem and ask how luminal androgen receptor triple negative breast cancer responds to CDK4 and 6 inhibitor after FGFR4 activation. Also, we want to check if activation of the FGFR4 with simultaneously inhibition of CDK4 and 6 affect the proliferation rate of luminal androgen receptor triple negative breast cancer cells. We started from the analyze the clinical data, and as you can see, the overall survival of breast cancer patients, depending on the level of FGFR4 expression, shows that the high level of expression of FGFR4 is a good prognosis for the patient. When we narrow the research group, we can see that there is some kinds of trend. Also, the most seenable trend is can we see in the luminal androgen receptor triple negative breast cancer patients. However, because of that narrowing, the number of research group is so small that we cannot assume it as statistic significant. So we go to the some wet party and use the model of triple negative breast cancer luminal androgen receptor cells and the condition like inhibitor of CDK4 and 6 and inhibitor FGFR4 rablotinib, Also, as a ligand specific for fibrillary growth factor receptor 4 is fibrillary growth factor 19. We start for the experiments to optimize the concentration of inhibitor, and also we proved that inhibition of both CDK4 and 6 and FGFR4 significantly reduce Cal-148 cells proliferation. Then we go to the 3D colony culture, and here we can see that if we use the same condition, the CDK4 and 6 inhibition significantly reduce Cal-148 colony size. However, the inhibition of FGFR4 did not reduce that size. And at the end of the presentation, I want to share the last of experiments and the results. We checked this if the CDK inhibition of CDK4 and 6 and FGFR4 activation in the MTTs assay, and as a result, active FGFR4 reduced the inhibitory effect of palbociclib. So go to conclusion after that few experiments. As you can see, the high level of FGFR4 expression is a good prognostic factor for triple-negative breast cancer, podtype. However, the active FGFR4 limits the effectiveness of the therapy with CDK4 inhibitors. And also, as you can see, that inconsistency could be caused by the microenvironment and other factors, which will be many of them during all the growing of the tumor. Thank you so much. Is there anyone interested? Want to ask some questions?
Okay, you mentioned something about there's inconsistency between the tumor microenvironment and the clinical result, the clinical trial data.
Yes, because basically on the clinical data, we can see that the higher level of expression of FGFR4 is a good prognosis. On the other side, when you look in the in vitro experiments, we can see that the effect of activation FGFR4 reduce the inhibition effect of treatment. So we have see the both way of that mechanism. We are going to see what is behind it.
But then isn't that a problem for the trial then? Because then, I mean, you got to get to the trial from the in vitro experiment first, right? So how does that make sense then? Like, how are we going to resolve that?
Because if we use the inhibitor CDK4, inhibitor for CDK4 and 6, they could be limited by the activity of fibrillary growth factor receptor, and we can also inhibit the fibrillary growth factor receptor by robitinib, and make a mix of the therapy, combine the whole two aspects, and then the first therapy should work without the inhibition.
Okay. Well, thank you, Wojciech. The next speaker is Ms. Karolina Dudzic, who will be talking about the impact of FGF7, FGFR2, signaling and IRS1 protein on development of resistance to anti-ER therapies in luminal breast cancer.
So hi everyone, my name is Karolina, and today I would like to tell you about my master project, which focused on the impact of FGF7, FGFR2 signaling and IRS1 on development of resistance to anti-ER therapies in luminal breast cancer. So first, I would like to tell you a little bit about luminal subtype of the breast cancer. It is one of the 4 subtypes. It's characterized with high estrogen receptor and progesterone receptor expression. It is also one of the most common breast cancer subtype. It represents 70% of breast cancer cases. And nowadays, we have anti-ER drugs like tamoxifen and fulvestrant. But some of the patients can develop the resistance to those drugs, so we need to find new ways of therapies for those patients. So first, a little bit about estrogen receptor signaling pathway. We have the classical one, which starts from the estradiol, which activates the estrogen receptor, which when it's activated, it goes to the nucleus, and there it is active as a transcriptional factor. We also have the alternated pathway, which starts from the microenvironment of the tumor, from the fibroblast, which produce the fibroblast growth factors, which activate the receptor, which later can activate estrogen receptor. And it is also possible that the IRS1 protein might coagulate the activity of estrogen receptor in this pathway. And it can also go to the nucleus and be active as a transcriptional factor. Normally, the IRS1 protein is the cytoplasmatic protein, which mediates the signal from the insulin receptor, but it is believed that here it might coagulate estrogen receptor activity. And our team showed that FGF can reduce the activity of the anti-ER drugs. Here the example is tamoxifen. And from those results, they decided to focus on the FGFs. So my hypothesis says that FGF7, FGFR2 pathway induces resistance to anti-ER therapies through the IRS1 protein, and the purpose of the research is to examine how IRS1 affects the FGF7, FGFR2 mediated abolishment of anti-ER therapy in luminal breast cancer cells. So first, all of the results were obtained for two of the luminal breast cancer cell line, T47D and MCF7. I have the wild type and the overexpression of IRS1 protein. So first I checked if I had the overexpression in those cells, and yes, I had. So later I decided to check if IRS1 overexpression will have any influence on anti-ER drug sensitivity. And here we see that the cells with overexpression have the colony size is increased. So we conclude that IRS1 overexpression decreased anti-ER drug sensitivity. Later, I decided to check inhibitor of IRS1, which is NT219, and it should degrade the IRS1. And here we see that it degraded IRS1 protein in both of the cell lines. After that, I decided to optimize the concentration of this inhibitor. So I performed the MTT test and 3D test. And we see that the IRS1 inhibition affected cell growth. The colony size are smaller. And for the further studies, I decided to focus on those two concentrations. So later, I decided to check if IRS1 inhibition will abolish FGF protective effect from anti-ER drugs. And here we see that if we add FGF7 to the colony size is increased, but if we add NT219, the colony size is reduced. Later, I checked it for the anti-ER drugs. Here we have the example with tamoxifen. When we add tamoxifen to the cells, the colony size is reduced. If we add FGF7 to the drug, we have the increased colony size, but if we add to this mix NT219, we have the reduced colony size. We also obtain exactly the same results for fulvestrant. So if we add FGF7 to the drug, we have the increased colony size. If we add NT219, the colony size is reduced. I also performed this test for the cell line with the overexpression of IRS1 protein to check if the IRS1 overexpression will change something. But no, we have exactly the same results. So if we add FGF7 to the anti-ER drug, we have the increased colony size. But if we add our NT219 inhibitor, we have the reduced colony size. And based on those two tests, we can say that IRS1 inhibition abolished FGF7 protective effect from anti-ER drugs. And at the end, I decided to check ER phosphorylation mediated by FGF7. So it is well known that if we add FGF7 to the cells, the ER is phosphorylated in the serine 118 and serine 167. And when it's phosphorylated, it is also active. And when it's active, it also is going to degradation. And because of this degradation, the anti-ER drugs don't have their target, so they don't work. And because of that, I decided to see if the inhibition of IRS1 protein will change something. And I added NT219 to the cells treated with FGF7. And we see that the adding of NT219 will abolish the phosphorylation or make it on the smaller level. So we can conclude that IRS1 is necessary for the FGF-mediated ER phosphorylation. And here are some main conclusions. So IRS1 inhibition reduced cells proliferation. IRS1 inhibition abolished FGF7 protective effect from anti-ER drugs. And IRS1 protein is necessary for FGF7-mediated ER phosphorylation and activity. Thank you.
Questions? Questions? Yeah, okay.
So first of all, I would like to thank you for this successful presentation. And actually, I have the two questions for this IRS inhibitor. So the first question is, could you explain why you choose this particular one? And maybe do you know if there are other inhibitors of the IRS?
Okay, so before we were focusing on NT219, our team was checking the other inhibitor NT157, but there were some problems with this inhibitor. It sometimes didn't work. So they checked that there is the NT219 inhibitor, and they decided to check it because it is used in the clinical trials right now, I think on the neck cancer or something like this. So because it is in the clinical research, they decided that it is a good target for us.
Okay, thank you.
Thank you.
Any other questions? Anybody? No? Okay. Well, thank you, Carolina. And the final speaker of the day is Ms. Gabriela Mishta, who will be talking about the role of the interplay between FGF, FGFR signaling, and DUSP activity in resistance to luminal breast cancer therapies. So,
hello, my name is Gabriela, and today I would like to present you my master project, which title is The Role of the Interplay Between FGF, FGFR Signaling and DUSP Activity in Resistance to Luminal Breast Cancer Therapies. According to the World Health Organization, breast cancer is the most common cancer among women and the second most common overall. It is divided into 4 main subtypes due to the expression of receptors. The most common is luminal A subtype, characterized by the presence of estrogen and progesterone receptors. Currently, there are several effective targeted therapies against breast cancer, such as tamoxifen, which inhibits estrogen receptor activity. However, their effectiveness is limited by the intrinsic or acquired resistance which a patient can develop during the treatment. There are several possible mechanisms of this resistance, and one of them is stimulation from the tumor microenvironment. This might involve signal from fibroblast growth factor receptor signaling, which can activate estrogen receptor independently of estradiol binding. This results in increased transcriptional activity of the results and cause the development of the resistance. Moreover, it has been shown that the DUS proteins, which are MAP kinase phosphatases, are potentially involved in resistance against breast cancer therapies. The current state of knowledge indicates an interplay between FGF signaling and DUSP protein's activity. The publication mainly focused on DUSP6 and its negative regulation of FGFR-derived signals. FGF attaches to and activates FGFR, and it causes the phosphorylation of MAP kinase ERK. ERK activation leads to an increase in transcriptional activity of DUSP6 genes, and as a result, DUSP6 proteins suppress the signal by dephosphorylating ERK. Our team was looking for genes which can be involved in resistant and regulated by FGFR signaling. RNA-seq analysis showed that DUSP4 and DUSP6 genes are top hits after cell stimulation with FGF7 and FGF7 with tamoxifen. Hence, the aim of my project is to investigate the interplay between DUSP4 and DUSP6 proteins and FGF-FGFR signaling in the context of resistance to anti-cancer therapies in luminal breast cancer. Moving on to the results, I conducted all my experiments on the MTF7 cell line, which is a luminal A breast cancer cell line. Firstly, I checked the level of DUSP proteins in stimulated cells. After cell stimulation with FGF7 and FGF7 with tamoxifen, the level of DUSP4 and DUSP6 proteins increased, while after using tamoxifen alone, level of these proteins decreased. I also verified the level of MAP kinases, which are important components of FGFR-driven cell proliferation and substrates for DUSP phosphatases. These results prompt me to derive MTF7 cell line with overexpression of DUSP4 and DUSP6 genes using lentivirus system. As you can see, the levels of proteins are higher in the cell line with overexpression, which confirms the effectiveness of conducted method. In the next test, I compared wild type and overexpression cell line in the presence of FGF7. As I mentioned before, those proteins dephosphorylate MAP kinases. Therefore, we expected to see a decrease in the level of phosphorylated kinases. Inconsistently with our expectation, the level of phospho-ERK didn't change, and surprisingly, the level of phospho-GNK is higher for the cell line with overexpression. These results would suggest that overexpressed protein is not active. In the next stage of the study, adhesion-independent growth test in agarose allowed me to check cell growth in the presence or inhibition of DUSP protein's activity. First test compared wild type and overexpression cell line in response to tamoxifen. The condition with FGF7 confirmed that this factor reduces sensitivity of cells to tamoxifen, which means it is involved in development of resistance. We hoped that overexpression would mimic this effect. But as it can be seen, overexpression colony size is comparable with wild type without FGF7. I also conducted this experiment using 3 other anti-cancer drugs, and the results were the same. Next test was conducted using BCI, which is a specific inhibitor of DUSP protein activity, also in combination with FGF7 and tamoxifen. In this case, FGF7 also confirmed its protective effect on the cells against tamoxifen. BCI addition significantly reduced colony size for each condition. This suggests that inhibition of DUSP proteins leads to abrogates the protective effect of FGF7. This experiment also was conducted using 3 other anti-cancer drugs, and the outcomes were the same. Based on the presented results, it can be concluded that FGFR promotes increase in DUSP mRNA and protein levels. Analysis of growth cells showed no effect of overexpression on involvement of resistance. However, inhibition of those proteins abrogates the protective effect of FGF7, and moreover, suppression of DUSP activity leads to cell sensitization to drugs despite the signals from FGFR pathway. To sum up, although overexpression of DUSP4 and DUSP6 genes in luminal breast cancer don't development in resistant cells, it seems that the DUSP proteins may play a role in FGF-regulated resistance to anti-cancer drugs. And that's all for me. Thank you for your attention.
Questions? Any questions? Yeah.
Thank you for your presentation. I would like to ask you if you know, or maybe do you have any theories why the overexpressed proteins weren't active?
Okay. It is complex issue, and I can give just one clear answer, but we don't know exactly the reason, but we can have some maybe possible explanation. One of them can be that the construct which was used doesn't work as we expected. And maybe another explanation can be that this overexpression is permanent, not temporary induced. So it can cause that in this cell occurs some additional changes in pathways that we don't understand.
Okay, thank you very much.
Any other questions? No? Okay. Okay, thank you, Gabriela. And with that, we conclude today's session. See you all tomorrow.